we're going to get started with the career panel. And so this section is career panels and then data perspectives and data analysis. They're both panel-based. And this one is, these both are very interactive. We're going to have people walking around with microphones. So please ask questions once they get through their introductions. And I hope we have a great conversation. I'm going to go ahead. I'm just going to leave this up. I don't think that's too distracting for anyone, hopefully. I'm just going to let you guys start introducing yourself. We have, well, I'll just introduce really quickly. Julie Hill, Julian Rading, Garrett Collette, and Thane Middlestad.
Great. I guess I'm in the middle, so I suppose I should go first. My name is Garrett. I'm a scientist at Bristol Myers Squibb at the 400 Dexter building here in Seattle. It's the old Juno Therapeutics building, if anybody is familiar with that. I started at a lowly routine analytics role at Juno Therapeutics, and then I've changed roles a few times since then. I now work supporting both autologous and allogeneic CAR T programs at BMS. Our current focus is on, or a major project of mine is donor selection. So determining and predicting which healthy donors are going to be the best allogeneic, that end up being the best allogeneic CAR T drug products. So there's a lot of biomarker screening and a lot of fun scale down modeling and CAR T production to do in the lab. I think that introduction probably suffices for now, and so I will hand it off to Julian.
Awesome. Hello, my name is Julian Redding. I head up the flow cytometry team at the Allen Institute for Immunology just up the road. My background, obviously I came from England a long time ago, but once I came to America, I was working in a cell therapy group for supporting phase 1 clinical trials at the NCI in Maryland. That's when I first got my exposure to flow cytometry. Though I really didn't like it very much. I was more of a functional cellular guy. My feeling was that I was more interested in what cells did than what they looked like. But over the next 10, 15 years or so, as I got more and more involved in flow cytometry, used it for functional serology assays originally, and then following dendritic cell differentiation and using that model for drug screening, I started to get more and more involved in cell sorting as well as flow cytometry. And now it's a major part of the work I do and I enjoy. So it's nice to be here with so many flow cytometry people.
I think this is on. Yeah, great. Thanks, Julian. I'm Julie Hill, and I want to just first say thanks to the organizers for inviting me and all of us here, and also for putting together this great meeting at such a good venue. So I know this was a lot of work, and thanks. Okay, so I work for Cytech Biosciences. I started out there as an application scientist, and my territory was Pacific Northwest. I live here in Seattle, so many of my customers are here, and I love doing applications with all of you guys. 2 years ago, I moved on to our customer education team, which is really great. Also, I especially like training. And so I think I'll tell you a little bit about my background. I first learned about flow cytometry by doing undergraduate research with Peter Rabinovich here at the University of Washington. And that is kind of what sent me on my path. So I guess I'll maybe stop there. Yeah.
So my name is Thane, and I've been in flow cytometry for about 15 years. I started as a lab tech at a CRO up north in Everett, formerly known as SMBL USA, now Alta Sciences. Then I transitioned to working at a flow core lab at the University of Washington under Michelle Black for about 5 years. From there, I then went on to field service engineer for Becton Dickinson for about another 5 years. And now I'm currently the director of the University of Washington Flow Cytometry Core Lab. And that's where I've been for the last 3 years. And that's my background. Now opening up for questions. Anybody?
Sure. Thank you.
I
Open dialogue, so feel free. Raise your
do
hand.
have a question. Looking back, what was the key decision or turning point that set you on your current path in cytometry, and what made you choose it over other options?
Suppose y'all can hear me, right? I suppose I can go first. I kind of fell into this role. It was very accidental. I was several years ago, I was applying to medical school, which didn't work out for me, as you can maybe guess, which is fine. And then I took the routine lab core, which I jokingly called lowly before, but I have a lot of kind of love and respect for that time in my life. That's where I first learned flow cytometry. I had some lovely mentors, some of whom are in the audience today, and fell in love with the ability to, I think, like the CAR T space is super interesting. It has really, I think, transformed the treatment landscape for a lot of heme malignancies. And so being able to work in the cutting edge was really fascinating, and being able to get my hands, really get my hands dirty and like actually figure out, what these cells were like, what are they doing, and able to kind of be able to make the connection myself between phenotypic characterization and actual function. And so, like I said, I kind of fell into this role and ended up using a lot of flow cytometry to characterize drug product. And as I kind of matured into my career and moved onto my current role, which is a lot more exploration and biomarker discovery and kind of telling a biological story using the data that we generate from characterization, I've graduated from conventional cytometry to spectral cytometry, building these large panels and using those to answer very real questions that we have on CAR T biology. It just holds a really special place in my heart. And I think also one of the number one things that I think sometimes people gloss over is the strength of the community. I think the flow cytometry community is really unusual in how tightly knit and supportive it is. And so I think the community aspect to it as well, as evidenced by all the people that are here today and all of the people that have conspired together to make this meeting a success, has really kind of kept me in the community. So that would be my answer.
Sure. Okay. So I mentioned briefly earlier dendritic cells. I was very interested in dendritic cells. Got the opportunity to be trained by this guy called George Stingel in Vienna, as well as bumping into Michael Lotes up at Pitt. Once I really got into understanding that the function and the important central role of dendritic cells in immunology, I kind of leaned on their experiences using flow cytometry to characterize particularly differentiation and activation. I then moved into drug companies where we were actually actively targeting therapeutics to either activate or suppress dendritic cells, depending on whether it was cancer or autoimmunity. From there, I got into sorting, and I had my FaxVantage, which was, if you remember back in those days, it was about the size of a room. It was amazing. I was the only person in the company that could use it. I became special for that technology. And then more recently, I've been able to, leaning a little bit on what Garrett said about the community, is we were able to build a team from scratch here in Seattle at the Allen Institute for Immunology. And I was very fortunate to stumble across these amazing young women at the moment, but Alex originally and Charlie too. So for me, it's the ability to be involved in technology, to be really excited about what I can see about a cell in terms of its activation status and its differentiation, but then also to work collaboratively with a team of early career scientists, and just from my perspective to see how amazingly smart and quick they are to learn things, and how kind of stupid I am, I feel in comparison. Back in the day, it was simple, but the way that these scientists that I'm fortunate to be surrounded with these days, it's really exciting to come to work.
Okay, I might be answering kind of a different question. I've worked in clinical labs, I've worked in research labs, I've worked in biotech here in Seattle. And at some point, I decided that there were some gaps in my cytometry knowledge, and I've been in cytometry most of that time. And so I was at a Cyto conference talking with Mario Roederer one day, and I said, hey, if I came to your lab for like 2 weeks, would that be okay? Could I come learn from you guys? And he said, 2 weeks? No, but if you come for a year, you can. And so I thought about that. And I went back to him and said, if you're serious, I'm game to come for a year. And he said, I'm serious. And so 1 year turned into 5.5. And that was an awesome experience. And so I guess what I would like to just say to people as maybe a recommendation, talk to people. If there's someone whose work you admire, talk to them. Maybe there's a place for you in their lab. So, and that also speaks to the amiability of people in the field.
Yeah. So for me, similar to Garrett, I fell into it. I had no idea what flow cytometry even was going out into college, coming out of college. So starting at the CRO, I was working in the small animal area. And then I wanted to transition into the lab, and that's where I first got introduced to flow cytometry. And uh, from there, it's just been going down that career path ever since. And this community here that is in the Pacific Northwest is very valuable for getting to know the flow cytometry area. So if you have any ambitions for whatever you want to do, reach out to people here, come to these events. Uh, uh, yeah, it's such a valuable resource.
Thank you.
And yeah, we're wanting it to be an open dialogue. So raise your hand. We have roaming microphones to come get you.
Right. Thanks, panelists. I guess I have a two-part question. The first part is, what makes you the most excited and engaged in your roles every day? And the second part is, what are you most excited about for your roles in flow cytometry and your roles specifically for the future?
Well, for me, it's helping out the users and solving the problems that may come up either with the instrumentation or with people's experiments. It's always fun trying to solve these puzzles that come up every once in a while. It can be aggravating it too, as well, especially if it's an ongoing intermittent problem and you're trying to rack your brain as to exactly is going on. But overall, it's very enjoyable work.
I'm going to say a similar thing. So I have always liked teaching. And so in my role as on the education team, I'm training people all the time. And I always learn from the people that I'm training. There's something they always teach me. Everyone knows something that I don't, which is great. So I would say for me, just helping to train people and also with a focus on helping people do the best science that they can. So that's kind of what I hope is my contribution for the future, is just helping people do really good work.
Yeah, I think right now the most exciting thing for me is just the way that the technology is blooming. We're very fortunate at the Allen Institute for Immunology. We have a benefactor in Paul Allen's foundation, so we're able to basically work with the latest and greatest instruments. What we've been able to do, as well as generate solid data sets and develop protocols that are actually robust and of relevance to other industry partners or research groups. So now people will come to us as a group and say, hey, we've got this thing, would you like to try it? So it's very exciting. Leaning into what Julie said, coaching and teaching is also a big part of it. But I think what I'd suggest all of you do if you get an opportunity is to talk to people in other omics, because flow cytometry was effectively the first single cell omic in terms of what we could do in terms of high throughput, et cetera, But now you've got CytSeq, you've got single cell RNA-seq, ATAC-seq. We're very fortunate again to be in a lab that we have folks and friends that do all of those omics. But for us to see where flow, because for a while you start to think, well, CytSeq is the new exciting thing. Flow cytometry is dead and buried, but that's not the case. As you learn more about it, you seem to find there's a niche for flow, there's a niche for sorting, and there's a niche for CytSeq. And once you actually start to get into that and see where my role would be, or what can I use this technology to tell me about a cell process, or can it have impact on patient health if I look at this aspect other than that aspect. And I think that to me is the most exciting thing about flow cytometry, is that it's not a standalone technology. It's part of a family of interconnecting technologies that really have the opportunity the potential to move science forward, and medical science of that, which is the end of the day. We're vested in patient health. We want to have impacts in the real world rather than, just writing papers, etc. That's my point.
I think adding on to the comment about kind of real world impact, for me, the most exciting part of the job is to be able to make medicine. I think that's that's pretty unique, and the ability to kind of make something that can like make drugs with your own two hands is really fun. I mean, I'm up here today talking about flow cytometry, but there's a lot of other tools that we use in the lab. We do all sorts of functional characterization. We do like generating CAR-Ts from scratch using scale down models. So to be able to make drugs kind of with your hands and then test them is super cool. And I think to be able, I don't want to beat a dead horse in terms of talking about cell therapy, but I imagine many of you are familiar, and maybe a subset of those are interested in the field. But to be able to inform, I think the production, like the life cycle of a drug. So like when you manufacture a CAR-T, you need to figure out in the manufacturing process, for example, how long to grow it out. How long can you grow it out so that you can, maximizing dose. This is just an example, but maximizing dose on one hand, and then compromising efficacy is the other hand. And so being able to like directly support the life cycle of a drug that's going to help somebody is really exciting. And we're doing that constantly at work, whether it's evaluating, doing donor selection, like I mentioned earlier, or figuring out what the best region is to activate your T cells before transduction. Those types of things are what we're supporting all the time. So being able to make medicine again, I think is a really exciting part of the job. And as far as the future goes, in the kind of old school cell therapy manufacturing, like in Breyanzi was the Juno Therapeutics anti-CD or like CD19 directed CAR. Our characterization strategy, which was very successful, was more simplistic than it is today. For example, we know that T cell memory has a correlation with efficacy, and so we might characterize our CAR-Ts using maybe a small memory panel, and then use that to be able to find links between process endpoints and what makes an efficacious drug. But kind of looking forward to the future, we have all these new tools at our disposal, like multicolor flow cytometry. We're using regularly some 30 plus color panels to characterize CAR-T product. And that I think just opens the space up a lot more to do biomarker discovery, and also thinking about like utilizing flow in novel and unique ways. Several years ago, I spoke on the topic of infinity flow, which is kind of a novel approach that was developed at Fred Hutch. And using that is like essentially a way to do like 400 color phenotyping of your samples of interest. And so there's like so many new and interesting things in the field that are being written about every week. So I'm really excited about future applications. That would be my answer to the question. Thanks. Yes, I think we have one here.
Hi, thank you guys for being here. I was just curious if you could talk a little bit about something that I think about in terms of like roles of managing a flow core or doing like lots of experiments with different groups, is the idea of like standardization of the assays between experiments and between labs, and how easy or hard it is to do in your institute. And I was wondering, particularly with respect to like doing research in more of academic setting versus more of an industry setting, or like how you feel that's easy or hard when you're managing different experiments or different groups.
For standardization, we've pretty much left it up to the users for standardization of the instruments, at least the instrument that they're using. And I recommend to run their own QC to make sure they're hitting their targets, similar to what Catherine was talking about, hitting those MFIs on a consistent day-to-day basis. At least for the academic side of things, it's the data analysis and the data robustness that I think can be difficult to disseminate that information out to all the different users and PIs within an academic setting. And I think within a more commercial setting, it's much more standardized, much more rigid, and easier to implement because you have to. Yeah. Anything to add for commercial or academic?
Yeah, we're kind of halfway house, but I gotta take a breath here because I don't want to insult anybody accidentally. But standardization, best practices are hugely important. A lot of our work has been longitudinal studies with patients over a 3-year period. So in order to string those data points together over 2 years, we really do have to do the same thing every time in terms of how we treat our flow cytometer. And it's not just making sure your beads hit an MFI, it's making sure that, I'm going to lecture now, but it's making sure that your flow cytometer is properly cleaned, properly maintained. And I tell you that the simplest thing that if any of you want to actually get an improvement in your data, it's your liquid handling that's the big weakness. If you can do a little bit of automation, Integra ViaFlow is great for us because everybody does their multi-channel pipetting a little bit differently. Some people flick their plates, some people aspirate their plates. I flick my plate a lot harder than Veronica up the back there. So as soon as you can get into something where you can standardize those variables, I tell you, you're going to be so surprised how much better your data is. And when your data is better, it's consistent, you publish and people actually read your papers and they can actually reproduce what you do. And that's how we have impact and power. It's not just us in our lab, it's being able to enable other people to do quality work just by following a few simple suggestions and guidelines. That would be my thing I basically say every morning to our friends at my work. So it's important for what you want to do in terms of data quality.
I just want to hark back to Catherine's talk this morning, which I thought was outstanding. And there's really, you can't put enough emphasis on instrument standardization, assay optimization, all the things that Julian's talking about, that Catherine was talking about. It's critical, no matter whether you're in an academic situation or a clinical situation. Of course, in clinical things, there's definitely more focus put on it, but if you want good data, you've got to do the work.
Yeah, from a more industry perspective, we think about data standardization, quite a bit. We're really lucky to have a really great internal flow core that does a lot of the standardization that the rest of the group has spoken about today in terms of setting MFI targets and then doing like regular cleaning and regular QC. And so I am personally fortunate enough to be able to mentally outsource that to somebody else, which is really nice. Would recommend that to anybody that can. But from I think another thing that this group hasn't talked about yet is a couple of approaches have really allowed us to make a lot of use out of our data. Number one is this is a very simple idea, but utilizing backbones for our large spectral panels. And so in the CAR-T world, we have 10 markers that we are always going to want to characterize no matter what. CD3, CD4, CD8, CAR, some memory markers, and so on and so forth. And so we know we're always going to be using those markers. And so we've been leveraging this idea of modular panel design and flexible panel design. wherein you like lock maybe a set of reagents and a set of channels for like the same colors every time. And this is helpful in comparing data from panel A to panel B. But the other kind of surprise benefit of doing this is really saving on reagent costs, because you're not, and like reagent management. We have like 4 30-color panels that we use in my group. And so antibody like inventory and management is really difficult, and making sure we have enough and that they're all titrated and they're all logged in the same way can be difficult. And so standardizing like the actual panel setup where possible and where it makes sense helps us a lot. And then also in terms of like reportable, another thing we think a lot about is reportable standardization. So in some of our larger panels, like, the number of reportables that you can spit out of your panel increases like exponentially by the number of panels you have, especially when you're characterizing T cells. There's like, a handful of markers you use to define major subsets, but then many more markers that you're using to define like T cell functional state, like activation markers, exhaustion markers, checkpoint inhibitors, so on and so forth. And you end up having this panel that's enormous and has like, we have like 700 reportables from one of the panels that we use regularly. And so we've worked like managing that and making sure that the data is always reported in the same way so that it's manageable from run to run has been like a big like point of focus for us, and focusing on that. And we have capabilities in our team to build like structured data inputs. So we have like these tools that kind of like align formatting and align reportable naming, so that we get, when we upload it, it's always the same way every time. We really get a lot of utility out of that, especially when characterizing samples from some of our trials longer term. So that's another way that we use standardization to realize some efficiencies. Hopefully that answers the question.
And I just want to take this opportunity to remind everybody how important antibody titration is. This is one of my favorite topics. I love titrating antibodies. I think I should start a business. Titrate your antibodies, everybody. Don't ever skimp on that part. Just a plug.
Yes.
I would like to hear, what are the skills that you find to be the most important in your profession at the moment? And a bonus question is, what is a skill that you found out that you didn't have, and how you overcame that problem?
That's a really excellent question. I think uniquely, I'll like speak just on my role, but in the industry setting, we're frequently not executing projects just within like our own group for our own knowledge. And I think this is true of many other people as well. But we have, there's like a lot of people working on cell therapy at the 400 Dexter site. There's folks that are developing assays, that are folks that are developing the manufacturing process. There are folks that are in the commercial manufacturing facility. There's a lot of folks to whom we're accountable. And so I think being able to like just work productively with a lot of different people, and being, this is like maybe a silly answer, but being enthusiastic and being kind of like a team player, and always being willing to kind of go the extra mile for kind of your quote unquote customers, your internal customers. That has reaped a lot of dividends for me, just by being the, one of the first people to volunteer to help, or to, if somebody, you have instrument time and somebody really wants to get on the instrument, but it's your time, to the extent, being willing to share and to help when necessary I've like got a lot of mileage out of that, because in return, I have many folks who are willing to help me in return. And so that is maybe like a soft skills answer to that question, but I have found that to be really helpful for me in the industry setting.
I think from my perspective, I guess the skill that I leaned on most, and I encourage amongst the team, and I would look for if I was rehiring somebody, is curiosity. I think in order to innovate, you need to be curious. And I think once again, I'm speak too highly of my team maybe, but that's one of our superpowers. I think is just being curious, not just being bored of coming to work, looking for something that you can do differently, or at least kind of thinking about what could I do differently. So that's a real power. And then kind of harping back to that automation thing, to me, working at starting to get into automation or understanding the relevance of automation, I always was kind of scared of that. It seemed to me that automation would be stealing jobs from people. But once you know how to implement it, it actually enhances what people do. It enables a summer intern to be able to do liquid handling as well as a seasoned professional. It actually enables and elevates. It doesn't actually replace. So that's from my perspective something that I've certainly learned in the Allen Institute for Immunology that I really was kind of naive to before.
Skills, that's right. Okay. Thank you. For me, the skill I use most at my job is the troubleshooting skill of knowing how to diagnose a problem and to solve it, whether it be an experimental problem or an instrument problem. That has been very valuable to limit downtime. One that I didn't know I had and probably still need to work on is just general like people management and like project management. That's there's always something to improve on. So that'd be my improvement.
Just to bring it another skill. So for me, communication, I feel, is quite an important skill. Just in back a little ways for me, after I wrote and turned in my thesis, I read it and went, oh my gosh, my writing is terrible. So I right away signed up for a technical communications program. I did a certificate program at the U. So things like that are other just ideas. If people are interested in business management or project management, those are things that would enhance for sure any work that you're doing in the lab.
I think I have it. I was hoping you all could talk about your educational background, and especially talking about some of the kind of non-traditional career paths and opportunities that flow cytometry has. There are more technical roles and more educational roles where you're not taking that traditional grad school path, and just wanted to hear some advice about that.
So for my background, I went to college for forensic science, biochemistry, biotechnology. Haven't really used any of it since then. But I think for me, what would be more non-traditional, and just get a wide experience and wide exposure to all possible avenues within where you're currently working. So like in a core lab, you have your engineers that you can that come in every once in a while, and you can learn from them at least the internal operating of the instrument itself. And then the people that come and use the flow core, you can learn more about the experimental and the biology side of things. Then within that can just branch out into other aspects like sales or being a software, either software engineer or application specialist. Trying to think of other like really non-traditional roles. Nothing is coming to mind. Anyways, that's something to add.
No, I can just tell you my education background. I graduated from Seattle U with a BS in humanities, but I always liked science and went on to the U and got a BA in humanities. I went to the U and got a BS in biology, and then went on and got a degree in toxicology. And I have a master's. I don't have a PhD. So so there's that. And I've made my way fine without a PhD. Although there definitely are some ceilings. Yeah, I'm not sure how to. There's plenty of different ways you can go. And Thane highlighted a number of the different positions that could be flow cytometry related, definitely engineering applications. Yeah. How much to
Sales. I'm going to say I'm a little bit different. I grew up in England, and I went to science straight from high school. I worked for SmithKline Beecham. So I high school education, working in a lab, a microbiology lab. Within the year, I had a patent and 2 papers, so I was doing okay. But the thing about England is that my company paid for my education, so they basically sent me to university 1 day a week and paid for it full. So I'm not like in America where you come out with your degree and a whole bunch of debt. Actually, kind of cool. By the time I got my degree, I had 6 years of industry experience, and then came to America. I was basically running a lab for the NCI. In immunology, I had no experience in immunology whatsoever, but I had an English accent and they seemed to believe that I was intelligent. And then fortunately, I once again, I surrounded myself with smart people, and we became a really powerful part of the NCI's drug development thing. In terms of additional education that you can do that's more technical or not necessarily a PhD, I would suggest every opportunity you have, take the manufacturer's training on an instrument rather than just being shown how to do a protocol by a friend in the lab, because you actually get the very basics and the real fundamental understanding of what you can do. So that's something I would strongly suggest, and we as a team, that's what we all do. But I think that's an opportunity. And then to what Thane was mentioning as well, when the engineers come in, hang out with them, see what they're doing, ask them what they're doing. Because I think, once again, it leans into curiosity. And with Thane was our engineer with our ARIA back in the day. Julie here was, even though she'd started working for Cytec, she had done a lot of work with the Symphony, which we just had. So I asked Julie to help us set up our Symphony. And then in return, we bought 3 Cytec Auroras over the next period of time. So it wasn't for free. And then Garrett came begging for some leftover expired reagents that we had, a Legendplex. And then a few years later, I think BMS took our PT1 T-cell panel that we'd built as part of a paper we released and used that as one of their backbones. So yeah, it's kind of cool. It gets back to community. We all kind of have worked with each other or know each other or have crossed paths with each other. And I'm sure that I know there's a whole bunch at the back that I know very well from other places too. So I think they're the 2 things I would say.
Yeah. And to add to, I just thought of something. So you can also look at the components of what makes flow cytometry. So you have optics, lasers, the optics and the lasers. You have the fluidics, you have the computer component. So if those are also interests of yours, you can go into those routes as well, like programming or like there's circuit boards in there. You can look at like fluid dynamics. Again, like working with lasers. Lasers are really cool. And just you can go into the various branches of what makes a flow cytometer or what makes flow cytometry.
Thanks. Yeah, my educational background is also just with a BS in biochem from UW, so also a non-PhD holder. And so far, I feel like that has not, thank God, has not really limited me in my career yet. I do think that there, as Julie mentioned, there is at some level likely some sort of, I feel at least that there is some sort of ceiling that I'm going to run up against at some point in my career. But I think especially on the West Coast, and also especially with some of the younger startups, at least in the cell therapy industry, I think that ceiling is going to be a lot more relaxed. And again, this is just totally anecdotal. But I found that, it's a little more, stodgy in some places, but a little bit more flexible in others. And I think even within an organization, you'll find some, I think, stratification with with respect to attitudes towards advanced degrees. So in like the early research, for example, I would imagine has like a kind of a stronger focus on like the real biology, the real kind of true deep exploration, versus the kind of like the development or the manufacturing setting, or kind of the regulatory setting. So there's like, multiple different places you can go in industry. And at least my sense is that those types of arms that are more manufacturing facing, you'll find a little bit more, I think, opportunity for advancement in those areas. So that would be, I don't know, my thoughts or my recommendations from an industry setting. And then to echo another theme that I think everybody has been talking about on the stage so far, the impact of personal relationships in your career growth, I think, like, cannot be overstated. Every opportunity I have ever got has been from just like talking to organizers or talking to other folks at conferences like this, or at vendor happy hours, or really wherever. So I like to think about this like when I was in like the mid 2000s. There's this movie with Jim Carrey called Yes Man. I don't know if anybody has seen it. Essentially, the premise of the movie is, as you would expect, he is kind of this like stodgy loner, but then he like decides to just say yes to everything, and his life really turns around. And I feel like that is kind of the ethos that I bring to kind of the, I don't know, the workplace and what I'd like to think about for my own career, and has worked for me personally. So those are my thoughts.
Okay, so this is our perspectives in data analysis, which we are going to have short talks from each of the speakers, and then with the remaining time, they have 1 hour. The remaining time, it will be open to ask them questions about what they're doing and why, and hopefully you learn lots of really cool stuff from them.
Hi, everyone. I'm Peter Morawski. I'm a faculty member at the Benaroya Research Institute. I've been doing flow for, gosh, I don't know, 20 years-ish. Love flow cytometry. I talked about the infinity flow stuff last year, if anyone was here for that talk. And I'll talk a little bit about time-dependent analysis today. So happy to be here. Thanks for the invitation.
Hi, I'm Sam Klebanoff. I'm a PhD student in my 6th year, also at the Benaroya Research Institute. Yeah, I've been doing flow cytometry pretty much my whole PhD, and actually even before that, I worked for a biotech company in the Bay Area called Cell Design Labs, where I also did a bunch of flow cytometry assay design. Yeah.
My name is Andrew Konechny. I'm also a graduate student in the same program Sam is. Our lab does a lot of high-dimensional panels, and I've been doing flow cytometry for the past 10 years. Today I'll be talking about the importance of batch controls and standardized gating.
Hello, my name is Hugh Macmillan. I'm a staff scientist in Evan Newell's lab at Fred Hutch, and I've been coping with some large cohort cytometry data analysis needs. And I'll talk a bit about one of those projects and one small subpart of this one project, and show just some of the nitty-gritty of the sorts of batch effect issues that can arise.
Hi. Okay, stay on. My name's Anna Koos. I also coming from Benaroya Research Institute. I've been doing flow for about 8 years, and mostly what I do is kind of within the clinical trial space. So a lot of that involves kind of large sample sets, high parameter flow, and I'll be talking about some practical analysis strategies in that context today.
So the first thing I think about with data analysis is how consistent is my data across runs. So I'm going to be talking briefly about the importance of batch controls and standardized analysis plans, or just how to set gates, because no analysis can change how good your data was when you started. A lot of members from our lab are presenting posters. Quinn, Nicole, and Alex all have one. So if any of these subjects interest you, here's the plug, go talk with them. And now we can get into the talk. So as a lot of us seen with these high dimensional panels, flow cytometry is quickly moving into big data analysis, where we have different programs to help us do dim reduction or clustering of our data. But a lot of our panels have some issue with this. And this is because of batch variation that occurs within our panels from run to run, where an important step in a lot of these analyses are normalize or somehow correcting for that. And if we just do DR or clustering on any of our data from runs, most of the variance we capture is just the change in MFI from run to run. And this is CyTOF data, but we've known this for flow cytometry too. If we stain the same sample over different acquisitions, we can have instances where a positive population has different thresholds for MFI. And batch variation can come from several sources. It can come from me, how well I pipette, as a lot of us have talked about today, liquid handling. It can come from the stability of our reagents in addition to our machine settings. And so I'm a practical person. What I want to talk about is how do I upfront account for any of this variation and either limit it or I'm able to pinpoint where it occurs. And Catherine today has done really well on talking about the first two kind of ways that we go about standardize and optimizing our machines. So I'm not going to cover those. However, some of our new generation cytometers do have some of these built in mind with their improved QC settings per day. What I'm going to talk about instead are batch controls or biological replicates and how they can inform me and the variation I cause in my experiment. And the important thing you need with a biological replicate is kind of two things. You need something that'll just be a lot of cells that you can run day to day to day. And you need to have an idea of how you're going to analyze that. And in particular, I'm just going to talk a little bit about how I inform where I set my gates. For a source of cells, our lab uses a leukopak. We can freeze down hundreds of vials of cells because we can get about 9 billion out of one donor. To illustrate the importance of batch controls and a standardized analysis plan, I'm going to be talking about this 28 color panel used for antigen presenting cells. This is an iteration of OMIP44 from our lab. And it looks at a bunch of different cell types, but then has phenotyping markers particular for antigen presenting cells. And this is kind of just all those fluorochromes and markers. But what we like to do is in our analysis, separate out our gating tree in two parts. The first is our lineage gating. How are we going to go about defining each of these cell subsets? So I'm not going to go over all of these individually, just that we have an idea of where each cell subset will pull out and when we'll go on to define other ones. And then for each of these, bolden cells that define a cell subset, we plan on looking at all of our different phenotyping markers that are in this panel. And so we have kind of that separate gating scheme that we'll use down here. This is what that looks like in action for our lineage gating. A lot of these are easy for placing gates, as lineage markers tend to be bimodal or have very predictive protein distributions. And so again, I'm not going to go over how we define a T cell, just that we have that as a predefined component of when we're building a panel. We'll then go and look at each of our cell types by their phenotyping markers that we have in this panel. And there are about 17. And I'm showing a histogram for each cell type from T cells all the way down to CDCs. And we can see that we have kind of a different problem with gating here. And that is that phenotyping markers tend to be more continuous, or because they infer different functions of our cells and they can change over stimulation or what have you. And so here I'm showing where I set my gates for my CDCs, and I've applied these to all my different cell populations. But we can see that there aren't just bimodal patterns that we have. We have something like here where there's a continuous, oh, sorry, my mouse isn't showing. Okay, that's okay. We can see things like CD85K where there's this continuous protein distribution. And so where we place a gate might be more difficult for that. So now I'm going to segue into tools that we can use for gating whenever we make these analyses. And our gold standard is the FMO. And this is where you have your entire panel, but you just leave out one of those markers so that you have a clear separation of where your positives are from your negatives. These are strenuous to make, so we usually reserve them for proteins that are either dim or on that continuous spectrum. The other, when we don't have an FMO available, what I like to use and others do also is something that I'll call an internal reference. It is a co-expression pattern that will separate out your positives. So here's a panel from Thomas Lichty, where he was using CD154, marker of activation, to pull out all these effector molecules in his stimulation assays. And so you get this nice L shape that shows your positive population, and then right below that, your negative population. So just another tool we use. Another type of internal reference we build in our panels are negative cell populations. So this panel, oh man, okay. Sorry. For this panel, we include T cells. They're going to be negative for most of our phenotyping markers. And so this just acts as another population that can represent any background binding that may be happening so that we can help place a negative threshold. Great, this one worked out. The last tool I like to use for helping me set gates are actually my single state controls. So when I run a panel, I mostly use cells for unmixing or compensation. And besides being a great tool for that, they can help inform where to set a gate based on spread. And so here I'm showing CD45RA, BV570 by IL1R1PE, and we can see that there's some spread happening into this channel. And so I can use this to help me inform where I need to place a gate, say if my cells are 45RA positive, where this doesn't occur in this other combination between CD45RA, BV570, and TCR gamma delta PECF594. If you are using cells for your single stain controls at the titers in your panel, another helpful tool for understanding how spread will affect your co-expression patterns is the total spreading matrix. This is similar to the SSM matrix. It just doesn't have the fluorescent intensity normalization. So it accounts for the spread that is happening in your panel at your fluorescent intensities. Now with all this in mind, I take all my standardized gates and I look at my biological replicate over time, and I can see consistency. And if there isn't consistency, I can pull out that run and say, okay, do I need to drop this from my analysis? So for here, we had consistency for all of our cell types, in addition to our phenotyping markers on our CDCs. And then the last thing before I wrap up is that you can also use your biological replicates to inform any of your normalization algorithms. CytoNorm uses that to help normalize your MFIs across your protein distributions. And I'll be done. So think about your standardization controls, as we talked about a lot today, whenever you want to plan one of these large analyses. And thank you.
Okay, next up. Do you guys know who's next? It's Anna. Do you want a pointer? Sorry, Andrew, I didn't grab.
All right, thank you, everyone. As I mentioned, today I'm going to be talking about some practical strategies for practical analysis strategies for large and high dimensional data sets. So I work in the Human Immunophenotyping Core at Benaroya Research Institute, where I analyze data from large-scale longitudinal clinical trials generated from high-parameter cytometry panels. In my time in the core, I've worked on upwards of 20 clinical studies, ranging from 30 to 600 samples per project, and have designed and executed panels with up to 37 parameters by flow and upwards of 45 parameters by CyTOF. So part of my job is to decide on an analysis strategy that appropriately uses the markers in my panel to answer the scientific questions at hand. And the two approaches we typically use for analyzing cytometry data are manual gating and multidimensional clustering. These are two methods that are well established, and their use cases can be broadly differentiated by the kinds of questions that they answer. So for more directed confirmatory questions, manual gating is typically appropriate. For example, in the study of the effect of CD2 depletion on circulating Treg and exhausted CD4 effector cells, we're asking a very direct question about specific populations that can be concisely gated and reported. On the other hand, multidimensional clustering is better suited for more exploratory or qualitative questions. So here we're asking how the composition of CD8 T cells changes in response to CD2 depletion, a qualitative question that lends itself to clustering. So these are two very similar clinical contexts. We're both dealing with CD2 depletion in a similar disease context. However, because the questions that we're asking are different, the analysis strategies that we're taking are different. And this is a very simplified example of how you might choose one strategy over the other. But in reality, these decisions are guided by more than just the nature of your scientific question, especially in a high throughput environment. So there's a number of other interrelated factors I consider when deciding on an approach. In addition to the scientific question, I'm also considering scale. So both in terms of numbers of parameters in the panel and numbers of samples in the study. I'm also considering reproducibility. So inter-study reproducibility, which is often really important for tracking biomarkers across clinical trials, as well as intra-study reproducibility in terms of consistent measurement across all samples of a study. And this is also related to scale. As the larger the study, the more potential there is for batch effects to arise. And also thinking about time. So, we all have deadlines. It's important to consider the urgency of, the data delivery. But it's also important to consider effort. So even if I had the time to, gate 500 samples 50 different ways, that doesn't mean that it's necessarily going to be worth my effort to do so. And then the last thing I'm considering is duration. So is this going to take place over 2 weeks, 2 months, 2 years? And that also, closely relates to both scale and reproducibility. And so there's attributes of each of these factors that influence whether clustering or gating is more appropriate. So I touched on scientific question already, but in addition to considering the type of question that you're asking, it's also critical that you assess the ability to answer your question. And to do that, you need to have a good understanding of what your populations of interest are and how you're going to define them. In terms of scale, the larger the scale, the more difficult and laborious it's going to be to manually gate all of your populations of interest on all of your samples. And this is, of course, related to time and effort. And clustering is much more scalable than manual gating. And then thinking about reproducibility, manual gating is inherently more subjective than unbiased clustering, and that can have effects on the consistency of measurement within a study, especially if it spans a long period of time. And then, on the flip side, if you're tracking a specific population between studies, it's really important that you define that population the exact same way that it's been defined in the past, which would typically require manual gating. And to add to this complexity, the importance of these factors is going to be project and context dependent, and one factor might outweigh the others in the decision-making process. So in my position, working with precious, irreplaceable clinical samples, making sure I'm using those samples appropriately to answer the scientific question will always be paramount, but my overarching goal is to produce high-quality, reproducible data while balancing the effort required to process hundreds of samples per study. And so far, I've kind of presented this as like a binary decision between clustering and gating. And if you're working with a small panel and limited samples, it would be appropriate to choose one or the other. But if you're working with panels that have like 30 or 40 parameters, it's likely those panels were designed to answer multiple questions, and choosing just one approach may not be sufficient to answer them. So what we actually do in practice is take a hybrid analysis approach where we're implementing both clustering and gating to answer diverse questions and balance needs within a single data set. So I'll briefly go through an example here of a study where I employed a hybrid analysis approach. So I designed a 35 parameter spectral panel to measure the effects of B cell depletion in 150 samples. And one of the main goals of this study was to measure the magnitude of B cell depletion and the rate of repopulation. So that's a very directed question and a metric that will be compared across other trials of B cell depletion. So here I'm gating and reporting, I'm manually gating total B cells and reporting that. But also of interest was the phenotype or quality of the repopulating B cells post-treatment. To get at this question, I chose to export my gated B cell population and cluster all B cells together. This was a more exploratory question that lent itself to clustering, but also B cell phenotypes are complex and not always the most straightforward to gate. So in clustering here, I'm saving myself a lot of time spent gating these populations on 150 samples and also increasing consistency of measurement within the study. So from the cluster data, I can export marker intensities from each B cell subset to understand differences in marker expression between populations and over time. And then one feature of our in-house clustering pipeline is that we can export FCS files with the cluster ID included as a parameter, which gives us the ability to further investigate these clustered subsets in Flojo or other software. So because in the study, we were specifically interested in which B cells are proliferating, and Ki67 is a marker that's traditionally gated rather than reported as an intensity readout, I'm actually going to go back to gating and manually gate Ki67 on these clustered FCS files. So here I'm showing an example of where I've started with gating, gone to clustering, and then actually gone back to gating to answer my question in a thorough but also kind of efficient way. And then to just continue on this example, this is the same project. Sort of a secondary aim to the B cell questions were the effect on CD4 T cells in response to B cell depletion. So here I'd hope to be able to take sort of a parallel response or parallel approach to the B cell analysis and use manual gating to define my parent population and then cluster to define CD4 subsets to see if they change with treatment. And at first glance, it looks like clustering kind of yielded all of the CD4 subsets that I would have expected, given the markers I used to cluster. But when I kind of take a closer look at this TPH cluster that I'm defining based on high PD-1 expression, it does look like there's a little bit, I don't know if it shows up, but there is a little bit of CXCR5 expression where I would expect none. So I went ahead and I guess investigated that further and exported the clustered FCS files from this clustering attempt. And what I saw was that these cells in the TPH cluster all express PD-1 and ICOS, which is what I would expect. But I'm also seeing a number of cells that are expressing CXCR5. And that suggests to me that I'm not able to cleanly identify this population of interest by means of clustering. So for that reason, the clustering is out. And I had to default to manual gating, despite the benefits that clustering might have provided in this project. So I also just want to emphasize that it's really important to make sure that you're able to answer your questions kind of above all other factors. Yeah, so just to quickly summarize, several factors guide selection of appropriate analytical approach. And these factors are project and context dependent, with the goal being to balance robust data generation with time and effort. Adopting a hybrid analysis approach can achieve multiple scientific and practical goals within large, highly dimensional data sets. And every data set is different. So it's really important to keep your scientific questions in mind and evaluate your results critically. So just want to give a big thanks to current and past members of the HIP core and the Long Lab and everyone involved in planning today's events. Thank you.
Thank you, Anna. And our next presenter is Macmillan, which is here. Oops, yeah. And second work for you?
It'll work for me.
Okay.
Not sure about the people in the back. We'll zoom in on a few spots. I don't know, does this work? No. Yeah, so my name's Hugh McMillan. I work with Evan Newell at Fred Hutch, and we say yes to lots of projects, I think. This is a project led by Stephanie Lee, who's pictured up in the top left. She's at Fred Hutch. And it's a huge number of samples, 750 or something like this, of folks from different studies, several different studies, but same T cell panel, same non-T cell panel, and all of them have some form of graft versus host disease in response to a treatment for their hematological disorder. 39 batches, all corresponding to the day in which 10 to 30 samples were run per batch. And the idea was to look at T cell and B and non-B cell subsets in connection to severity grades for a variety of different tissue contexts, to understand how GVHD is manifest in different tissues and whether the severity has any relationship to what's going on in the blood, what's going on in the PBMCs. So in this bottom left panel, each color is a cluster. Each little vertical bar is a sample, and we have decomposed each sample into the relative proportion of cells from that sample that landed in a cluster. And so this is just showing the CD8s. We'll do these, kind of divide and conquer a big project like this by gating out CD8s from all 39 batches, gating out CD4s, gating out B, gating out non-B, and running 4 separate parallel clustering streams. And so this diagram illustrates this process that we've got. It's quite an iterative process, and at times can seem a little endless, perhaps, to my collaborators who think I can just press a button and give them clusters, and the clusters will never change. You know, there won't be 30 one day and 29 the next. Each panel here, in this box plot on the far left, each panel is a cluster, and the x-axis is a gradation of the skin severity GVHD response. And so we're looking for downstream whether we can generate all of these clusters and come up with some meaningful biology and some statistical discovery. Thankfully, I'm not tasked with actually doing the downstream statistics. This is Ninjing and Tim. Tim Randolph is in the top row there, and Ninjing is below him. They're in Arnold building there at Fred Hutch. And Tim is actually an applied mathematician, not really a statistician. He's somebody I can talk to, and he kind of appreciates. He doesn't run away from these outputs of these clusterings, and understands some of the processing, that pre-processing and post-processing of the data that becomes iterative here. So what we're doing is dividing and conquering, right? So we'll have 39 batches. We cluster each batch separately, and then we cluster the centers of all the batch-specific clusters. So that's this meta-clustering step. At that level, at the cluster centroid level, we can apply various off-the-shelf batch integration, batch effect treatment methods. But a lot of times the reality of the data takes these off-the-shelf methods kind of off the table. And it's nice to just know exactly what you're doing with the data when you're transforming it to address these perceived batch effects. It's a combination of data visualization, sort of qualitative assessment of how well we're doing, as well as some quantitative assessment. And there's issues with both of those approaches. So this is a pretty polished clustering here at the bottom, and it's something like 30 CD8 subsets, which is quite a lot in my experience. There's a lot of panels that you start to get up to 15 to 20 CD4 clusters or CD8 clusters, and they don't, it's over clustered just by looking at it and saying, you know, these clusters don't look that different, right? So you can be more objective than that, but right. So I just wanted to share some of the perspective on this sort of data arts and science, really. So what I'm looking here, I'll zoom in on this, but I'm thinking about treating batch effect as really understanding when we think there's a real true difference between cells, right? So cytometric, right? We're looking at the distance between cells here. And when we're transforming our data, what we're essentially doing is changing the sense in which we're measuring the distance between cells, and we're doing that in a batch-specific way. So let me just zoom in a little better here. So now each row is a batch. It's a day in which a Cytec Aurora run was made out there in Bethesda at the NIH. And in this first row, this first column of this first panel here, you can see quite a bit of wobble. There also. Those are just the 3 repeated controls. The y-axis is CD45RA, and I've just grabbed a handful of the parameters for the different x-axes for the different. So this is just a subset of the parameters. It's all the batches. On the left of this panel is controls. On the right is the samples. So I just want you to see how the first column, the repeated controls and the samples, you can kind of see the same sort of wobble, right? So if you're going to use whatever nudge you need to push this over to align your controls, it's going to have a nice effect on your samples and align those samples. And that's what's happened over here on this other panel, is that I've just used I've aligned the repeated controls and then applied that batch specific nudge to the samples and gotten them to align. And so the clustering looks better because now the clustering doesn't resolve batch quite so easily, right? And I can kind of ramp up my clustering resolution and see a little more. So this is just a even more zoomed in on just a few parameters and just some of the early batches. And I've highlighted, I guess in the next slide, in red here, you can see again on the left, just the repeated control. On the right, it's samples, and then gray is the collective repeated control. And so I've highlighted these first 4 or 5, I guess 5 batches, because this one parameter, ROR GT, ROR gamma T, there's a bit of a shift in the controls that we don't really see in the samples. And so we do this correction based on the controls. It seems to work great for a lot of parameters, but there's a couple where it doesn't so much. But each one of these batches was pretty large. It's pretty well mixed. And so opted to use the samples themselves. So this is raw, unadjusted. Here we've aligned using the control samples, and here I'm just aligning using the samples themselves, taking the median of within each batch and aligning those samples. And so that's an example where, you know, sometimes you need to kind of balance your faith in these controls, maybe not apply blindly just what the controls are doing to the samples. So that's one little messy vignette. Another one is dealing with this sort of spread, particularly in the negative realm of things. So if we're talking about the distance between cells, 20 dimensions, 23 dimensions, right? The square root of the sum of the squares of all the component differences. So there's this whole thing called the curse of dimensionality, right? Where you're just getting a number, distance between 2 cells, and you've got 20 dimensions, things get a little blurry. And so you don't want to send your clustering off chasing distinctions between 2 cells that aren't, or differences that aren't really a real distinction between those cells, particularly when you have this negative spread. You don't really want to have your distance between 2 cells be coming from variation in the negative realm of a value. And so you can do a little compression of that signal, a little smooth compression of that signal. So this was a little trick, just creating a little transform to smooth things out. So this would just map a range of values, a range of arc-cinch transform values, 3 to 6 into, what is this? Why is this there? Maps -3 to 6 into -2/3 to 6, right? So I'm just doing a little compression in the negative realm and squeezing together these values for a given parameter to not have clustering chase those distinctions. And this is maybe when I knew we were sort of cooked with AI, right? Because I drew this picture and I wanted to correct what I was doing, make sure it was, you know, correct. Well, I knew it was correct because I'd implement it, but I thought it'd be fun to see if AI could solve it. So I just gave it a picture, and sure enough, it gave me all the coefficients that my spline needed to be to have this nice sort of smooth transition, and gave me the code to plot that. But another little AI vignette is, you know, one thing that happens is you're constantly repeating these clusterings, and my collaborator wanted to finalize them, you know, wanted to label them. And so, of course, I came up with a new clustering, and I needed to apply their old labels. And I thought, oh, well, surely I can just upload these pictures or upload the little data frame that went into this picture, and get it to label it the way this human labeled it. And I spent all day on that, and it kept failing. So there's still some role for us humans to play. So those are a few little vignettes for you.
I love the use of AI for things like that, where you take a picture and you're amazed what we can actually do with it. We could have a whole, I think, conference just on what AI is doing to science, but that'd be a different one than today. Okay, we are on to Sam. Yes. And if we run over a couple minutes, we're just going to run over a couple minutes, but I want to make sure you guys get your slides.
Pretty quick, hopefully.
Okay, you want your pointer? I'll give it to you anyways. There you go.
Thank you. Okay, hi everybody. I am realizing that I forgot to make a title slide as I was hearing all these other lovely talks. But just to reintroduce myself, I'm Sam Klebanoff. I'm a 6th year PhD student in the lab of Daniel Campbell at the Benaroya Research Institute. And what I wanted to talk about briefly today is just a really quick troubleshooting vignette to do with sample barcoding that I thought might be interesting to some people in this audience. But just to start by giving a very quick and broad overview of my PhD project, what I'm looking at is the memory T-cell response to cytomegalovirus, or CMV, which is one of the most common viruses in the world, and it's a latent virus, right? So you can imagine that the viral load when you're infected with CMV, there's sort of an initial lytic stage of infection, followed by a long-term latent stage where the virus is kind of periodically attempting to de-silence. And as a result of these kinetics, the T-cell response against CMV does this really interesting thing that we call memory inflation, where you get a boost of effector T-cells that help in clearing this initial lytic stage. And then after that pool contracts to a small pool of memory T-cells, what we actually observe is that pool of memory T-cells increases in size constantly throughout life to really staggeringly large percentages of the memory T-cell compartment in humans and mice. And so what my project has been focusing on is sort of sampling these CMV-specific T-cells at different time points throughout this process and looking at cells specific for different viral proteins. And so in the kind of the first half of my PhD, I was doing a lot of sort of high dimensional flow cytometry, including infinity flow. And in the second half of my PhD, I've transitioned a little bit more into doing single cell RNA-seq and single cell TCR-seq and more bioinformatics style approaches. And I'm happy to talk a little bit more about that experience during the panel. But for today, as I mentioned, I'm just going to go through a really quick vignette about sample barcoding. So as I mentioned, we're sort of interested in sampling T cells specific for different viral antigens at different time points. And so it's kind of a lot of different types of samples that we want to look at all at once. So sample barcoding is really important in my project. And when I was developing a lot of these assays using a flow cytometer, we came up with the strategy, and Peter actually helped a lot with this, but we came out with the strategy using 4 different CD45 fluorescent antibodies here, which I'm labeling 1, 2, 3, 4. And you can see this is a population of PBMCs that I split 15 ways and gave each a unique combination of these 4 fluorescent antibodies, right? And you can see some of them, every cell is either positive or negative for the first 2 in this middle graph here, and then the second 2 fluorescent antibodies in these other 4 graphs. And when you do a UMAP analysis, this is on a whole fluorescent panel, which was a 30-color panel. You can see all 15 of these populations distinguished nicely from each other. But where we ran into a bit of a pickle was in moving a lot of my analysis from this high-dimensional flow cytometry approach to more of a single-cell RNA sequencing approach. And we really wanted to maintain our ability to barcode lots of populations and run them simultaneously. But we ran into this issue where, the strategy that I had developed was using CD45 antibodies as barcodes. And in the case of our single-cell RNA-seq and single-cell TCR-seq, the issue that we had run into is that the populations that we wanted to barcode, the only way to get those populations apart from each other was via sorting, right? Because we use fluorescent tetramers to identify our different populations, and then we want to barcode them separately before sequencing. But the problem is, if you want to get good quality TCR sequencing data, you have to really, really minimize the time between sorter and sequencer. This is something that we learned through hearing about experiences of other groups at BRI. And so we were not able to sort our populations of interest and then label them with our CD45 site-seq antibodies. So what we ended up doing is coming up with this strategy where this is an example of one experiment where I'm looking at T cells from 2 different time points from a donor. And we have 3 different specificities of CMV-specific T cell that we are interested in sequencing from each of these time points. So what we did is we, starting from each of these 2 vials of PBMCs, we enriched for CD8 T cells and then stained with the fluorescent sorting panel that I was using, as well as the CytSeq panel, with the exception of the CytSeq barcodes. But after doing that sort, I then pre-split each of these tubes into 3 tubes each, which you can see here, where each of these 6 tubes now is labeled with a unique CytSeq barcode. So we used 6 anti-CD45 total seq-C CytSeq barcodes here. And then after I had sort of pre-labeled all 6 of these tubes, before I had done any sorting, what I did is I essentially took the tube labeled with 1 and the tube labeled with 4 and combined them. Same with the tube labeled with 2 and the tube labeled with 5, and then 3 and 6. Each sort of got recombined. And then these are my 3 different specificities that I'm sorting, either tetramer negative cells here or tetramer positive cells for 1 of 2 CMV viral proteins. And so basically what I was able to do is I was able to sort those 3 populations from these tubes and then basically go from that step directly into the sequencing approach. And because I had done this approach of splitting and individually labeling the tubes before even getting on the sorter, what you end up with is, from this tube, I have tetramer negative cells that are labeled with barcode 1 if they came from the early time point, and they're labeled with barcode 4 if they came from the late time point. Right, from this middle tube here, we have PP65, which is a viral antigen, PP65 specific cells that are labeled with barcode 2 if they came from the early time point, and 5 from the late time point, et cetera. You can sort of see how this works, right? And at the end of the day, this approach ended up working really well, right? So this is just looking at the CytSeq data in Loop browser from this RNA sequencing approach. You can see here these 6 populations are the 6 individually barcoded populations that I talked about before. And these little grayed out islands that you can see around some of these clusters, those are the clusters that were not uniquely identified by a single barcode, right? They had somehow gotten stained with more than 1 of our CytSeq barcodes, but you can see that's actually a relatively small percentage of the sample. So overall, this strategy worked pretty well. So if anybody else is struggling with a similar problem, hopefully maybe that is useful to someone. But yeah, that's all I got.
Great talk. That was really interesting. Okay, we have our last speaker. Peter, where are your slides? Wow, that's fun.
There she is.
All right.
So we're a little short on time, so it's kind of appropriate, I feel like, that we're actually talking about time as a variable in flow cytometry. And I couldn't help but kind of throw in a nod to maybe the most stable fluorophore of all. So I think the point is that if you're going to do one thing before you start analyzing your data, which is where we've talked about downstream analysis things from the other individuals in the panel, I think time is really one of the most important things. And I wouldn't just rely on the little button in FlowJo that tells you if you do or don't have a nice sample according to time, the little bubbles that's there, because it might tell you that you have a bad sample if you just have, you left the tube to run dry for the last 10 seconds. And you can deal with that, right? We can gate around that. So this is fun. If you actually look back to the history of this, 1987 is the first example I could find where this is great. I love this. Using time as quality control. This is straight from the abstract. By hooking the computer clock directly into the acquisition logic and replaying fluorescence or light scatter versus time. And this is, we know that this is to this day still an exceptionally important part of our analysis. And this is a really nice example of what simpler times it was for flow cytometry. So another way of viewing that is saying, what do you actually look at when you're looking for time, right? And I really hope that people are doing this because it's one of the first things that I try to teach my new trainees when we talk about how to analyze flow cytometry data. The air bubble, for example, right? We know that bubbles in the fluidic stream, right, in the core stream is going to actually disrupt not only the stability of the signal, but the fluorescence signal itself, right? And we need to be able to account for these sorts types of things. And then pressure instabilities, right? We know that under fluidic control, we need to keep stable pressure over time, especially when you're sorting, but in all cases under pressure. And what we know is that, we have those 5 lasers or however many lasers you have in your machine, and they're focused through these prisms into a narrow space, and then we have laser delay, right? And so actually, when you have pressure instability, the further away you are from that sort of reference laser, which is normally the blue 488, the more disrupted your signal can become, right? And so these are things that both, well-maintained and QC'd instruments that people have talked about today, but then also your attentiveness as the downstream analyst for where these things might be an issue. And of course, I didn't really talk about it, but you can also see in this example here, sometimes, especially when the sample amount in your tube gets very low, the pressure can change, right? And you might want to actually make sure that you gate out the tail end of this acquisition cycle. Similarly, you want to be very careful about waiting to actually record your sample for a few seconds after you start acquiring it to deal with that pressure buildup that happens early on. Okay, I'm going to give a quick anecdote here for something that we used this for that's based around fret energy. I'm not going to go into the details of this, but we know that things like time and temperature and oxidative stress can impact floors, right? And how fret energy works. And we see this not just in flow cytometry, but in other forms of imaging, as well, imaging mass cytometry and fluorescence microscopy, photo bleaching, all of these different things are things that we need to be aware of when we're dealing with fluorescence antibodies, fluorescently labeled antibodies. Okay, so here's my one anecdote. We actually published this a few years ago in Immunohorizons, just as sort of a, please be careful when you're doing these certain practices in your sorting. Essentially, so my lab studies T cells, specifically in the skin, and we were sorting these different populations of cells. And one of the technicians that I was working with had this, beautiful panel that we had developed to actually look at different populations of memory and conventional and effector T cells. And within 5 minutes of sorting the cells on the fusion, the BD fusion, we actually started seeing some things that looked pretty strange. So CCR10 PE, for example, suddenly shot through the roof while we were acquiring. Well, that looks really weird. What's going on, right? And the AF700 signal had completely gone away. And my technician was really worried that she had done something horrifically wrong. And some things looked relatively normal. So it wasn't really sure what was happening, but there was some kind of bizarre things happening, right? And it turns out, long story short, is that a combination of the light inside of the chamber on the fusion and the fact that we were using a formulated serum-free media for our T cells was the problem that we had encountered. Essentially, there was an oxidative stress reaction that was killing some of our floors and not others. And so the sort of punchline of the story here is that we were able to replicate this by actually turning on the LED light in the chamber in this essentially cells that were in serum-free media. And within seconds, some of our tandem dyes like PE-Cy7, and some of our non-tandem dyes like AF700, which is interesting, and I'm not sure I fully understand the chemistry of that, were getting destroyed. The PE-Cy7 signal, as I showed you on the previous slide, devolved into PE, and all of a sudden we saw this huge increase in PE, right? And other signals like this base fluor 421 were completely unaffected. What we found is that we could rescue this phenotype by putting either protein, BSA or FBS, or by actually quenching the reaction with vitamin C, which is sort of a known redox negative regulator. And this is sort of an example of all of the different places across the visible spectrum in our panel where we could actually see that oxidative stress changing the floors. So I think this is just like a nice set of data that suggests looking at time as a variable is a really critical thing. People have been doing it for a very, very long time. And I think especially in multi-parameter, high dimensional space is a really, really critical thing to keep in mind. If folks want to actually read more about the story itself, I just threw a QR code on there. So sometimes people are squeamish about taking their phones out, but by all means, please go ahead and grab that QR code. And I'll just thank Sam, who is the technician that worked on the project, and some of the people at BRI Cell and Tissue Analysis Group that helped support this work. Thanks very much.
So the next speaker is Eva Newell. He's a professor of vaccine and infectious disease division at Fred Hutch. Dr. Evan Newell is an immunologist who develops and employs new technologies for accurately identifying specific biological signature of human health and disease, including cancer and infectious diseases. He and his team work with blood and tissue samples using mass cytometry and other single-cell analysis methods to better understand how the specificities of immune T cells influence their roles in clinical productive response against pathogens or cancer.
Yeah, thanks so much for this really nice honor, Michelle, and everybody, the organizers, for this meeting. It's been fun. So yeah, my title, I'm going to tell you about how I'm trying to, or I'll talk about a bunch of different things, but I think one of the unifying things that we've been working on recently is to try to improve our coordination of immune profiling efforts. And I think flow cytometry is an important aspect of this. And I think we've heard today a lot about how important it is to really standardize or work towards standardizing flow cytometry data so that you can link, collaborate better across different trials or even within trials. But from my perspective about what we've been trying to do with flow cytometry, I think pretty much everything I've been doing throughout my career has been some sort of flow cytometry in a sense. And we've mostly been working on humans, so we're trying to understand or trying to take advantage of the samples that we can get and really understand what's happening with the human immune system, based in spite of the fact that when you take a blood sample or a tissue sample, it's hard to know what cells in that sample are actually relevant for the disease or cancer or infection or vaccine that you're trying to study. So we've been trying to figure out, better ways to identify relevant cells, either through broad profiling, comparative analysis, or through analyzing antigen-specific cells, and in particular, antigen-specific T cells. And so the things that I've done throughout my career have focused on that are sort of related to this mission of analyzing immune cells in different contexts are one, to decode the high dimensional cellular phenotypes of cells, particularly T cells. And you saw Hugh talking about some approaches for looking at large flow cytometry data sets. But historically, we've also tried to work on new methods for just understanding high dimensional cytometry data. And we've applied this to understanding the diversity of tumor infiltrating T cells. So what's interesting is that everybody's in cancer, when you look at a tumor, different tumors can vary dramatically in the composition and phenotypes of T cells. And we've been working on this and particularly focused on trying to understand how the specificity or the tumor reactivity of T cells contributes to those phenotypic differences. And in fact, there are phenotypic differences between tumor-specific and also highly abundant bystander T cells that can infiltrate tumors. And so sort of leading from the tumor into the blood, we've also been really interested in trying to identify relevant T cell populations in the blood that can help us predict outcomes for cancer immunotherapy, but also in the context of vaccine and infectious disease. And so in both of these contexts, we're trying, again, to focus on antigen-specific cells where we'll have a better chance of identifying, relevant differences between responders and non-responders or different outcomes. And then, I'll talk about how we've been using peptide MHC multimers to study antigen-specific T cells. And this has been very useful, and we have some great ways of doing this. And you can really learn a lot about antigen-specific cells if you can identify them in a direct way using tetramers or multimers. But more recently, we've also been trying to look for other ways of doing this that don't rely on exact epitopes or even HLA associations. And so we've been starting to use TCR sequencing more and more to analyze antigen-specific T cell responses, catalog them, and track them. So that's something else that we're quite interested in. Not a topic for today, though. So the things I'm going to talk about today, in light of sort of that background, one is this Fred Hutch Innovation Lab that I also scientifically direct at the Fred Hutch, and I just wanted to tell you about what this does. And although most of what it's doing is not specifically flow cytometry, it works a lot with flow cytometry and has benefited a lot from flow cytometry for many of the things that it does. And I think, in relation to different assays that are being developed, it's important to link these things. And so we'll talk about how we're trying to, de-risk new approaches for cellular profiling at the Fred Hutch. And then I mentioned peptide MHC tetramer staining, and this has kind of been the core method that we've been using over the years. So I wanted to tell you some examples of how we use multiplex peptide MHC tetramer staining with CyTOF and how we're trying to learn from these examples and then apply, look for correlates using similar T-cell profiles in other studies. And then, as I mentioned before, this goal of trying to coordinate immune profiling, I'm going to talk about an example of clinical studies for CAR T-cell therapy, where we're using a variety of methods, kind of in collaboration across different groups. And the idea is to try to apply these types of approaches across other trials to gain insights across trials. But at first, we're showing sort of what we can learn by using multiple sort of platforms on the same clinical study. And then it's not really cytometry, but I wanted to tell you about how we're using 10x xenium, so spatial transcriptomics. And to me, it's kind of getting into cytometry because with spatial transcriptomics, we feel like you can actually segment cells pretty accurately. And so I'm going to tell you about a segmentation algorithm that we developed and that I think can be used to really get cellular information from these archival FFPE samples. And again, I think it's synergistic with flow cytometry of tissue and blood samples from the same patients. Okay, so the Innovation Lab. So this is a small lab that was started several years ago, actually initiated by Stan Riddell and the immunotherapy IRC at Fred Hutch. And the idea was to sort of de-risk emerging single cell and other immunotherapy relevant cellular analysis methods. And so what the lab has been doing, and this has actually been led especially by Anna Els, who's the lab manager of the Innovation Lab, what she's been doing with her team is sort of help try out new things that come along. So we've done a lot of beta tests for single cell sequencing platforms, spatial transcriptomic platforms, and other things. We've also done bulk TCR sequencing. And what we try to work with other researchers at Fred Hutch to get them started with new things. And then when possible, try to do comparisons across different platforms to look at pros and cons. And then our goal is to sort of make these things easier for labs to adopt and also move them over to shared resources to further facilitate uptake of new methods. And so just a plug for one of the things that we've done is this paper that is, I don't have the reference on here, in bioRxiv, but also this NAR genomics and bioinformatics and genomics, where we've sort of did this comprehensive comparison of single cell transcriptomics approaches. And we talk in this paper sort of the practical aspects in terms of how these different platforms compare. And hopefully it's useful for people considering using these assays. Oh no, there's something wrong with the slides. Okay. One of the ongoing projects also that we're doing, so yeah, downstream of this, we've worked on other new emerging assays. An example for this is a Flex version 2, which is a much higher throughput version of 10x Flex. And so we're really excited about this. This has been used at the Allen Institute especially, so we're working with them on some aspects. And so this is kind of the latest thing that we've been testing out with a number of different groups. I won't go into too much detail here, except to say that it's quite a remarkable development in terms of the throughput of single cell transcriptomics that you can do. So we're really excited right now about getting this into people's hands. And actually, one of these projects with whole blood will relate to what I'm going to talk about later. The other thing that has been really rapidly developed, I think, by the Innovation Lab was the use of 10x xenium. And I'm going to talk about this later with respect to cell segmentation. But we actually did a number of pilot projects with xenium now almost 2 years ago. And these projects have all really been highly productive with papers and grants. And so by doing this kind of sort of early access to new things as they come out, I think we've helped a lot to get people using new methods and then sort of getting it going for many different projects. And that, again, was the vision for what the Innovation Lab is supposed to do. So we're open for input in terms of what we should be looking at next. And then also we have a Twitter blog, we have an internal blog. So get in touch with the Innovation Lab if you're interested in trying something new, or you feel like maybe we can share our experience with the assays or platforms that we've been working with. Okay, so next topic is about the multiplex tetramer staining, and this sort of relates more to the general introduction that I had. And so, when I started my lab, my vision was to sort of try to understand the high dimensional phenotypes of T cells in humans across tissue and blood samples. And then in order to do that, I wanted to relate it to the antigen specificities of those cells. So if you know the antigen specificities of any of these, highly diverse T cell phenotypes, then you can understand really what those phenotypes mean. And then vice versa, you can understand sort of what's happening for that response to a particular antigen. And so the goal has been to sort of broadly look at, high dimensional profiles of T cells using high dimensional mass cytometry or flow cytometry, and then in parallel look at specificities of cells however possible. And the main thing that I developed as a postdoc was this multiplex tetramer assay that works really well with CyTOF, and essentially we can combinatorially encode up to hundreds of different tetramers with combinations of heavy metals. And we make these cocktails of tetramers that we stain cell samples with. And then with the combinatorial encoding, we have remaining channels to do the profiling. So we have 20 or 30 antibodies combined with, 100 or so tetramers. And so this is not a super easy thing to do, but we've gotten good over it over the years. And so we run CyTOF on these blood or tissue samples, and these antigen-specific cells can be hard to find. But then when you do find them, you can then go and characterize their phenotypes. And so, for example, if you take a blood sample, this is a CD8 T cell sample from a healthy donor, and we can make a UMAP, describe sort of the range of phenotypes for CD8 T cells. And then this person had a bunch of different hits for antigen-specific, virus-specific cells. And then you can go and see that different epitope-specific T cells occupy distinct regions of that UMAP, and try to understand what those, antigen-specific T cells are doing, and also sort of what those regions of the phenotypic space represent. And so, again, we've been using this for a variety of different things. And so I wanted to talk about, and so I'd mentioned before we'd looked at this in tumors, but then what I've wanted to really do is understand how we can take blood samples and then understand what's happening with immune responses in the context of, cancer trials or other types of things. So a major goal has been to try to look at tumor-specific T cell responses in blood. However, the challenge for antigen-specific T cells is that it's really hard to predict which epitopes are involved, and even when you know which epitopes are involved, T cells in blood that are specific for tumor antigens can be extremely rare. And so we needed to find contexts where this would hopefully be a little bit easier. And so the two cancers that I'm going to talk about today are nasopharyngeal carcinoma. So this was a long-going project. So I'll go through some of the data for that. So NPC is an EBV-driven cancer in the head and neck. And so the nice thing about EBV is that we know a lot about T cell responses to EBV, and there's known dominant T cell epitopes that we can use to make tetramers and then identify those EBV-specific cells. And so the obvious question in NPC is what's happening with EBV-specific cells when you have an EBV positive cancer? And so can we learn about what tumor-specific T cells are doing in the blood by analyzing EBV-specific T cells in the context of this EBV-driven cancer? One thing about NPC is that it's not very responsive to cancer immunotherapy, so that's kind of something to take into account. And that's another question, like why is it that EBV-specific cells, even though they're there, are not controlling the tumor? And in addition, when you treat NPC patients, the responsiveness to anti-PD-1 checkpoint blockade immunotherapy is quite poor. So those cells are not really inhibited in the same way as for other cancers that do respond to checkpoint blockade. And a really amazing example for the opposite is a Merkel cell carcinoma. And as a lot of you probably know about this, there's a lot of research being done in Seattle on Merkel cell carcinoma. And this is, big consortium led by Paul Neum. And so we were quite lucky to connect with Paul Neum and use this opportunity to analyze T cells specific for the Merkel cell polyomavirus. Because in Merkel cell carcinoma, 80% of the patients have a Merkel cell polyomavirus positive tumors, which means that the virus itself is driving the cancer. And here again, these viral antigens are expressed by the tumor cells and then can be used to study the tumor-specific T cell response if you can identify those Merkel cell polyomavirus-specific T cells. So another goal that we've had is to try to compare Merkel cell carcinoma to NPC, two different very different viruses and very different virally driven cancers. And the other amazing thing is that MCC is highly responsive to checkpoint blockade, with more than half of the patients having actually a complete response to immunotherapy. So anyway, for the NPC work, this was work led by Nandita Kumar and then also taken over by Rachel Zemek, a postdoc. And the cohort that Nandita has analyzed is a cohort of 51 new diagnosis NPC patients. And the reason why this work has taken so long is that we've, in addition to the mass cytometry data that I'm going to talk about here, we also collected a number of other modalities and have had to work pretty hard at integrating these data sets. But I think for the question of what's happening to EBV-specific cells in the blood of these patients, the CyTOF data was kind of what we wanted to do. And so Nandita made a CyTOF panel that has 35 phenotypic markers. And then she also had, using multiplex tetramer staining, 56 different tetramers with a lot of focus on EBV epitopes. And she generated this really nice data set where she was able to detect many examples of antigen-specific T cells across these patients, including many examples of EBV-specific cells from these NPC patients. And by sort of, again, having many batches to run these 51 samples, she was able to integrate the data into one UMAP of CD8 positive T cells that sort of shows, in this case, like an aggregate of these 51 patients' CD8 T cells, in addition to some healthy donors that we ran in parallel. And, just to orient this UMAP, I'll show a few times. We have naive cells and then these sort of terminally differentiated effector cells, as well as activated and also these CD103 positive T cells down here, which are of interest. And so when you overlay the phenotypes of EBV, CMV, flu, and other antigen-specific cells, you can see that they occupy, somewhat distinct, but also somewhat overlapping regions of this UMAP. And just by comparing the healthy donors and NPC patients, you can already see that there are regions of this plot that are enriched in patient samples. And Nandita did a bunch of cluster analysis to identify this cluster 11 cell population that was specifically enriched in NPC patients. and also enriched within EBV-specific cells from NPC patients. So whereas other EBV-specific T-cell phenotypes were kind of mixed between healthy donors and patients, this cluster was particularly, sort of unique to patients. And in addition, she found that the frequencies of cells that had that phenotype also correlated with markers of tumor burden. So the patients that had this EBV DNA titer, which is kind of like a circulating tumor DNA quantity in the blood, and also a marker of tumor burden, tended to have a higher frequency of cells with this cluster 11 phenotype. Both when you look within EBV-specific cells, but also in bulk amongst total CD8 cells. So this phenotype was something that we were sort of happy to discover as a marker of something that's relevant to something happening in cancer in this case. And so by using those EBV-specific T-cell phenotypes, we're able to kind of zoom in on this phenotype of interest. And to me, it was interesting because these cells showed an activated phenotype. So they express activation markers like CD38, Ki67, or CD71, and also exhaustion markers like CD39 and PD1, as well as CD103, which is this marker that we heard about earlier today that is typically seen in intraepithelial resident memory T cells, but also in blood. About 1% of CD8 T cells express CD103, and these are thought to be recirculating resident memory T cells. So they were at once resident within tissue, and then as they recirculate in the blood, they've maintained CD103 expression, but they've lost CD69 expression, which allows them to leave tissue. And so interestingly, what we see for these tumor-specific cells is often a population that express a combination of an exhaustion marker, which is indicative of like sort of chronic antigen stimulation, and then CD103, which we think is indicative of recirculating out of tissue and presumably out of the tumor. And so she also used single cell sequencing to dig deeper into this phenotype and showed that these cells express other genes associated with tissue residence, exhaustion, and genes like CXCL13 that are known to be associated with tumor reactivity. And so for this work, I mean, there's a lot more that has been done in terms of correlating these phenotypes with other measures that we've done on the patients. But the interesting thing is that we found that this sort of tumor-related phenotype are activated T cells, but also activated T cells that express markers associated with exhaustion and recirculation. And other papers in lung cancer have also shown some, anecdotal examples of neoantigen-specific T cells that have similar phenotypes, including CD103 in the blood. And so I think that it could be that when you're looking in blood, one of the major sort of phenotypes that you have for tumor-specific cells are these sort of recirculating exhausted cells. So then just switching over to the Merkel cell carcinoma side, so this project sort of happening in parallel, but we managed to publish this work already. And in these papers, what we showed together with Paul Niem's lab is that actually the frequencies of Merkel cell polyomavirus specific cells that you detect in blood are very low. So even though it's a different virus, and so therefore I think T cells are primed in a different way. And in patients that have virus positive cancer, even then they have typically quite low frequencies of cells. We had one patient with quite a high frequency, but they're higher than the patients that have this virus negative tumor. But what was striking in our experiments for multiple cohorts is that the frequencies of the virus-specific cells are higher in the patients that go on to be complete responders. So having virus-specific cells at baseline before being treated with anti-PD-1 is actually a good predictor of response to therapy. So implies that you need to have some kind of tumor-specific response that's in the blood as compared to the partial responders or the non-responders. And also interestingly, over the course of treatment, in the complete responders, in fact, as the tumors respond and shrink, you lose those cells. So the source of antigen may be disappearing, and therefore we're not seeing these recirculating or tumor-specific cells. And this is just to show that you can actually use, if you can detect these virus-specific cells at baseline, you can actually predict survival of these patients in therapy. But what we were also interested in is the phenotypes of those virus and tumor-specific cells. And so this plot is showing a few markers that we zoomed in on, again, kind of informed by the NPC work. Comparing, in this case, these Merkel cell polyoma virus-specific cells with other antigen-specific cells. And you can see, similar to what we saw in NPC, CD39, so a marker of exhaustion was higher on these tumor-specific cells in blood, and in some cases CD103 as well. But then CLA was a marker that was also abundantly expressed on these Merkel cell polyoma virus-specific cells. And so based on the fact that each of these markers is actually quite rarely expressed on T cells in the blood and pretty low frequency, especially in healthy donors, we wanted to see if we could use a combination of these markers to predict outcomes for Merkel cell polyoma virus among bulk T cells. And in fact, if we use this marker that we looked at before, the combination of CD39 and CD103, this predicted actually a poor outcome. So the patients that have higher frequencies of these sort of what we think of as recirculating exhausted T cells in blood were more prone to not respond to therapy and progress more quickly. But in contrast, if we looked at cells that had the CLA marker, which was more common among the Merkel cell virus-specific T cells, that phenotype actually correlated with the frequencies of the virus-specific cells, which we knew was sort of associated with response, and then also predicted good outcomes. And so to me, the interesting comparison here is between two different contexts where we're able to look at tumor-specific cells in blood. In one case, we're seeing this phenotype that is sort of associated with X tissue residence, so these recirculating CD103 positive cells for the most part. So there are some other activated cells which may be sort of similar to what we're seeing in MCC, but this is still remains to be determined how they predict response to immunotherapy in this context. But in Merkel cell, we have tumor-specific cells that tend to have this other phenotype of CLA expression, and those are, the strong predictors of response. And so in terms of what we can look at in blood, I think this is important because we want to try to find ways when we go and do immune profiling of cancer trials or other things to know what kind of markers should we include in our panels in order to learn what's happening to T cell responses. And at least from these tetramer experiments, we're able to sort of zoom in on some interesting markers that we've been including in our panels. And the idea that at this point is that maybe this CLA phenotype, which we also showed to be a little less terminally exhausted in that they express TCF1 to a higher degree than these CD103 counterparts, may be more indicative of sort of an ongoing response. So let's say T cells that were primed and are sort of trafficking to the tumor, in contrast to these sort of CD103 positive, more terminally exhausted T cells that may be leaving the tumor and actually more indicative of tumor burden. So that's where we're at. There are kind of hypotheses at this point in terms of whether or not these markers can be predictive in other contexts, but at least it's something to try to look for. I have a couple examples. Don't worry, I still have quite a bit more to do. I have a couple other examples where we're similarly looking at antigen-specific cells in the context of other cancers. So we have a paper that was led by David Glass and Carolyn Shasha looking in the context of myeloma, and this is also in collaboration with the Allen Institute, where we sort of see the lack of evidence for a chronic, terminally exhausted T cell phenotype in myeloma. And this is in contrast to other solid tumors. And so basically the point of this paper was to say that, these hallmarks that we've come to learn from other tumors are not as often seen in myeloma. And we also used antigen-specific T cell profiling to sort of support those claims. And then another project that that we're using tetramers for is in the context of graft versus leukemia. So if during stem cell transplant for leukemia patients, one of the important mechanisms for clearing tumor or residual tumor is having the grafts of the donor T cells that can react with the tumor through mismatches in minor histocompatibility antigens. So these are genetic differences between the donor and the recipient that T cells can be specific for and actually can clear tumor. So this is kind of the original version of cellular therapy. And this work being led by Marie Bleakley, where she's characterized many of these minor histocompatibility antigen epitopes. And this fit well with our system where we could go and make tetramers for many different minor antigens and then screen for T cells that are specific for these in the context of transplant. And actually, we found a few really nice examples of these, SNP specific T cell populations. And then we were able to sort of track their frequencies and phenotypes over the course of transplant. And, I'm not going to get into too many details, but it's interesting that these, in this case, tumor-specific T cells that are elicited by transplant have many of the same features that we see for tumor-specific T cells that I just talked about. So, for instance, they express activation markers, CD39 and exhaustion marker, and also in some cases CLA, which is kind of interesting for leukemia. And this is just showing, again, using actually Hughes' clustering analysis method to sort of quantify the phenotypes of these tumor-specific cells over the course of transplant. And so this work is ongoing, but I thought it was a nice example of, again, sort of how we're ongoing uses of tetramers. Okay, so the next part is about sort of this sort of strategy of taking a trial and working together with different labs to analyze samples in multiple ways. And this is where we're really investing more effort in developing flow cytometry panels that we hope to be applicable to many trials. And we started this with a CAR T cell therapy project. And so the question for this project is, what are the correlates of toxicity? So this is, in fact, in the context of large diffuse B-cell lymphoma that is quite well treated by CD19 CAR T cells. And what happens is that this can be associated with a number of different toxicities. The two that we're going to talk about here are cytokine release syndrome, or CRS, and also neurotoxicity, or ICANS. And in fact, CRS can be handled pretty well with blockers of IL-6 and with steroids, but ICANS is harder to manage, and it's a big question into the mechanisms of ICANS, and also can we find biomarkers that predict ICANS so that these patients can be treated more aggressively during their therapy. And so for CAR T cell toxicities, the thinking is that CAR T cells go in and are destroying the tumor, and in the process they're secreting a lot of cytokines, interferon gamma, and things that activate monocytes and macrophages, and then you get this sort of cytokine storm that can cause CRS. And then similarly, these cytokines can induce leakage to the blood-brain barrier and then lead to neurotoxicity. And so there's a lot been done looking at PBMCs and the influence of CAR T cells on monocytes and macrophages, but actually much less is done with granulocytes. And in fact, in the early time points post-CAR T cell treatment, after lymphodepletion, there aren't very many cells in blood. In fact, most of the lymphocytes and leukocytes, for that matter, are very low in frequency. And so that's another reason why we started using whole blood. And then we're able to look at granulocytes. And so I'm going to focus also on trying to ask the question about the roles of granulocytes in CAR T cell associated toxicities. And so this big sort of collaborative effort involves multiple grad students, Nikita, Tony Chur, samples and sort of questions from Jordan Gauthier, and Sylvain Simone has been working with Jordan to assemble these cohorts over the last many years. And so the goal that we've had is to do a combination of things. So I'm going to talk about our efforts using smart tube buffer fixed whole blood and whole blood profiling. Spectral flow cytometry on PBMCs, which we're developing a panel for, and I'm going to talk about that. And then there's also been a lot of single cell sequencing, TCR sequencing, and then serum cytokine analysis in collaboration with Heather Gustafson at Seattle Children's. And for the PBMC panel, this is kind of an interlude. We've been working with BD to develop 50-color PBMC spectral flow panel based on that OMIP 102 that Andrew, Florian, and Martin Prilich have developed so nicely. And this paper is just a really good source of information about how to do this well. And so working with Aaron at BD, we've actually developed this big 50-color panel that is tried to be as simple as possible, given that it's 50 colors. So we only have surface markers, and it's quite broad, looking at all different T cell, NK cell, B cell, and myeloid cell subsets. I won't go into too many details, but just to say that it's working quite well. We can run PBMCs and gate on all the populations that we're interested in. Especially the T cell subsets that we're particularly interested in. And then it makes these, amazing UMAPs that you can interpret and see. And so we're using this to, compare different trials. And I'll also mention that we left 2 channels open in this panel so that we can include markers such as markers for the CAR T cells or other things that are particular to each of the trials. So the strategy is to have something that's relatively simple but extremely broad that we can run across many different trials. This is just another UMAP. And so we're actually using this for other CAR T cell trials. We've run it on HIV and acute infection studies. We're using it in the context of malaria vaccine and challenge studies, and also related to the graft versus host disease work that or somewhat related to the graft versus host disease work that Hugh was talking about. But now I'm going to switch back to CyTOF and whole blood. So for this CAR T cell trial where we're looking at correlates of toxicity, this cohort that are sort of described in this swimmer's plot is showing the time points that we've gotten from these patients relative to when the patients that have ICANS or neurotoxicity have developed neurotoxicity. And so the goal of this project was then to look for, whole blood cellular correlates of toxicity. And so from this whole blood panel, again, using CyTOF, we were able to sort of delineate a really broad range of immune cells from whole blood. And as expected, most of them in whole blood are granulocytes and neutrophils. And so we were able to get this, big cluster of neutrophils. And one of the sort of interesting things that pops out of these data when you compare the healthy donors to these, lymphoma patients at baseline prior to lymphodepletion and prior to CAR T cell therapy, already the neutrophil phenotypes are strikingly different between healthy donors that have a kind of sort of homogeneous neutrophil population and these patients that have had all kinds of other treatments have very diverse neutrophil phenotypes. And so this was a lead that we could, at least try to look for what this correlates with respect to toxicities that occur. And so we used, Hugh's approach to cluster and metacluster these many batches of samples that we've run. And the 2 sort of streams, as he calls it, were the sort of C66B negative or sort of mostly mononuclear cells. and then CD60B positive granulocytes that we analyze separately. And as I mentioned, at the pre-infusion time point, the composition of these healthy donors, so these stack bar charts sort of show the composition of all these clusters for the two different types of cells. You can see that the healthy donor controls are strikingly different from the patients that have these diverse and sort of aberrant neutrophil populations, and also more diverse and heterogeneous or new populations of mononuclear cells. Yeah. And so then, we've used some statistics to try to look at what populations are different over the course of treatment between people that go on to have ICANS and in the context of ICANS versus the people that don't have ICANS. And we identified a few different cell populations that are significantly different between severe neurotoxicity and not. And one of the striking ones was this monocyte cluster 19, which is actually a CD163, CD14 positive monocyte population. So it kind of looks like a monocyte that's differentiated into a kind of a suppressive macrophage. And you can see an example of a zoomed in part of the UMAP that shows that at peak ICANS especially, that population becomes a really dominant population. And this is just showing that there may even be some differences prior to treatment in the frequencies of those cells that might predict the onset of ICANS in these patients. So that's the monocytes, an example. And then in the neutrophils, we also see a lot of dynamics in terms of these, weird neutrophil populations. And the thing that's striking out or is showing difference to the greatest extent are clusters of neutrophils that have kind of an immature phenotype. So they express CD10 and CD101 to different levels. And then in addition, we also have some activated looking neutrophils that don't discriminate between severe and non-severe ICANS, but also sort of coincide with these toxicities that have also expression of HLA-DR. So it's not shown on this one. And so sort of in collaboration with Heather, we've looked at cytokines for all these patients in parallel. And from the cytokines, you can also see a number of differences between patients that have neurotoxicity versus not. And so then the interesting analysis is then to look at how, in this plot, what we're looking at is how the cell populations that we've quantified for each patient correlate with the cytokines that we've quantified for each patient, and then also how that associates with whether or not there's toxicity. So like these cytokines here correlate strongly with toxicity. And then these cell populations also correlate with toxicity and also correlate with these cytokines. So we can find these kind of coincident activities of cell populations and cytokines that can sort of associate and predict toxicity. And then a way to sort of formalize this analysis is using these immune modules that David Glass in my lab developed and actually published as part of another study looking at biomarkers for checkpoint blockade. And what he does is sort of group together features of cells, or in this case, cytokines, that have strong correlations with each other. And then these become these immune modules. And then he uses that to sort of reduce the dimensionality or the number of features, and then can more accurately identify correlates of outcome. And so for this, we've identified a number of these immune modules that contain the cell populations and cytokines that I talked about, and are kind of using them as, correlates of outcome, and to try to get some sense of how these things are working together to drive toxicity. So for this part, yeah, we've sort of done longitudinal profiling of CAR T-cell patients experiencing CRS and ICANS, and then we've sort of identified high dimensional populations of both monocytes and neutrophils that correlate with cytokines and also these toxicities. And so hopefully this study is an example of how we can use these kind of hopefully relatively standard panels that just look broadly and can find things that correlate across these different omics that we're analyzing, and then could be used in multiple trials. Okay, so the last bit, just in the last few minutes, I want to tell you about why we've been so excited about Xenium and spatial transcriptomics, and how I feel like cell segmentation has made it possible to use spatial transcriptomics as a way to analyze cells from these archival FFPE tissues. And so I'm sure you've been aware of this, but Xenium is one of these in situ spatial transcriptomics methods, which uses hundreds or even thousands of probes that can, anneal to transcripts within cells. And then there's these sort of combinatorial and amplification methods that they use to then image exactly where those individual transcripts are at subcellular resolution. And so what you're left with is an image with dots, and each dot represents individual transcripts. And you can measure up to thousands of different transcripts at subcellular resolution. And the challenge is that then when you have all these transcripts, you need to figure out, what cell to assign them to. And so at the time that we started using Xenium, the out-of-the-box method that 10x Genomics provides is this, very crude method, which basically identifies nuclei based on DAPI staining, and then expands outward until you run into another cell that's expanding outward. And so it draws these boxes around cells, and any transcripts that fall into these boxes are assigned to those cells. And as you can imagine, it's going to be that cells are going to have much more complicated morphologies, and transcripts that you identify could easily be misassigned. And so as Daniel and other folks in the lab were analyzing these data, it was a real challenge to sort of deal with the notion that there's a lot of misassignment and cell segmentation issues. And so Daniel developed this method that he calls ProSeg, or probabilistic cell segmentation. And the way that this works is it actually uses the identities of the transcripts to help inform how the segmentation is determined. And so it doesn't have any sort of a priori knowledge that goes into it. It just starts with cell boundaries, and then it adjusts those cell boundaries using something called a cellular POTS model, which is a way to simulate cells. And basically, he can use these algorithms that these cellular POTS models are based on to refine the boundaries of cells in a way that it tries to make the contents of each cell relatively uniform. So if a transcript is in let's say a B cell transcript is in a T cell, that B cell specific transcript would kind of seem abnormal. And so it would try to sort of adjust the boundaries to exclude transcripts that don't sort of match the other transcripts within the same cell. And it iterates this until it kind of gets to boundaries of cells that kind of have uniform distributions of each of the transcripts within the cells. So there's more to this, and you can read about it in his paper. But the downstream effect from my perspective was that it made the UMAPs look a lot more interpretable. So this was one of the original 10x UMAPs from a data set where we have this big smear, and we can annotate cell populations, but in fact, we know that there's a lot of sort of missegmentation and smearing going on. In contrast, Bayesian, which is another method that has similar principles to ProSeg, was able to much more strikingly separate cell populations. And then ProSeg is the algorithm that Daniel developed. But then the advantage of ProSeg over Bayesian is that it's computationally much more efficient, and also the sort of premise behind it makes fewer assumptions. And it leads to a more, normal number of expected number of cells. So this one inflates the number of cells, and also we think kind of misassigns cell populations as well. And so the bottom line for all of this is that this ProSeg method can take Xenium data and sort of make it into single cell data that you can better interpret. It's quite computationally efficient. And then he's also went on to sort of benchmark it against a number of other methods to show how it's able to identify the genes that of interest. And so, this is important when you're going to go and ask questions about what's different about cells in one location versus another. And so in this example, he tried to look for, what genes are different in tumor-associated macrophages versus macrophages that are further away from the tumor. And what comes out of this type of analysis typically are, missegmented tumor genes, right? So genes like that, he's labeled red here as spurious genes. And so the goal of his algorithm was to sort of identify genes that were, different between tumor-associated macrophage and get as many of those as possible, but not identify these sort of spurious genes. And so that's kind of a way that he was both benchmarking ProSeg and also showing how it is, as of yet, the most capable method for identifying, relevant genes in this kind of differential gene expression analysis context. And just as an example for what we've done with it, we analyzed some kidney tumor samples. So and actually there's been so many data sets run with Xenium that we've been analyzing this way. But in this case, we ran 4 different RCC samples. And then you can, what's nice is that you can see that the tumor cells from each patient are quite different, and that's expected for cancer, given that they're sort of driven in all different ways. Whereas the stromal cells and immune cells were all kind of overlapping between the samples, and then they have interpretable phenotypes. And then after you label them kind of using regular single cell annotations, you can go in and see where these cells are in the tissue. And this is just an example where we had quite a lot of B cells, sort of these large tertiary lymphoid structures, which were exciting to see. And then we were also able to sort of ask questions that we were interested in about T cell subsets. So Xenium is not very, the resolution can be not so great given that you don't get that many transcripts per cell. Nonetheless, we were able to zoom in and more or less parse out T cell subpopulations. And then further sort of analyze how those T cell populations, differ with respect to their association with tumors. And so we were curious whether these, T cells that have gene expression profiles associated with tumor reactivity were also co-localized with tumors. And for some of the tumors, they were. And so the ones that have a lot of T cells, we did see that these sort of exhausted looking CD8 T cells had significant co-localization to tumors. So that's kind of an example of what you can do with this kind of data. And then further, we're also able to, use these kind of spatial analyses to discover other genes besides CXCL13 that were associated with being close to tumors. So anyway, I think, this is not a single cell transcriptomics meeting, but I think to me, it's kind of in the same vein. So I wanted to mention that. Okay, so in total, I knew it was a whirlwind, but I told you about the Innovation Lab and how we're operating, and so please interact with us. I told you about sort of how I started my lab and what we've been doing in terms of analyzing antigen-specific T cells. More recently, we're moving into using TCR sequencing more instead of tetramers, but we're still highly active in using multiplex tetramer staining. And then, I'm really excited about much higher throughput spectral flow cytometry and being able to use whole blood to analyze samples from clinical trials. And my goal is to try to, coordinate these efforts a bit. And then also kind of stemming from the Innovation Lab, our experience using spatial transcriptomics and how I feel like it's another way of analyzing immune cells in tissues. So tons of people involved with all this work. Here's a list of some folks in the lab. I mentioned some of their names along the way. Many collaborators for all this work. The Innovation Lab, Anna Els is really the big driver there. For the spectral flow panel, which we're really excited about, it's been great to work with Martin's lab, and Andrew has given us a lot of advice. And then, of course, BD and Aaron Tisnick to help us design that panel. And yeah, some other collaborators for the different cancer systems that I talked about. So thanks a lot for your time, and yeah, looking forward to chatting over a beer here pretty soon. I know I went a little bit long, so I don't know if there are questions or if, okay, we do have, yeah. Maybe we take a couple. You can yell.
Great talk. Thank you.
Thank you.
I was going to ask, from my understanding, at least severe CRS is a risk factor for ICANS. Did you see if there was any overlap in some of your findings for what puts a patient at risk for ICANS and CRS post CD19 CAR T therapy treatment?
Yeah, definitely good question. And yeah, Hugh, you could talk to about this. He's been kind of in the thick of it. I mean, it's true that, I mean, the ICANS is a little bit later. So we'd aligned the analysis based on the peak of ICANS, and that's where we've gotten more significant differences. But yeah, it's hard to deconvolve because CRS happens, you know, kind of in parallel, right? And so indeed, I think a lot of these things correlate with CRS as well. And so I think, yeah, to me, this is also a good question. I think we've had better luck in terms of identifying correlates of ICANS. Also because in this cohort, I think the CRS is better managed, and so the CRS is a little bit lesser. And also I think what we're measuring is confounded also by their ability to suppress it with treatment, whereas with ICANS it's not as much suppressed. But yeah, I think these things come together, and I think that I mean, at this time scale, they're very close. And so I do think a lot of these correlates are there in both cases. Yeah.
The next speaker is Rafael Villasmil. He has led the flow cytometry core facility at the National Eye Institute for nearly 20 years, overseeing cutting-edge cytometry platform and supporting research in immunology, ocular disease, and clinical applications. An experienced educator and trainer, Rafael specializes in best practice for flow cytometry and quantitative single-cell analysis. Currently, Rafael is pursuing a doctorate in bioinformatics and computational biology at George Mason University, integrating computational approaches with empirical methods to advance health research from the lab to clinical applications.
Thank you very much. It's an honor to be here. The last 2 weeks have been an exciting time to plan this talk. As you know, I'll be talking about precision in location and timing. So you are all thinking submicron particle analysis, right? So the ideas that I will be discussing today are my own. They do not represent NIH, Department of Health and Human Services, or sponsor any of the products or services that I might or might not be mentioning over here. So what is this message that I am trying to bring to you? Well, it's basically that there is a lot of value on flow cytometry data that happens in the nanometer scale. It has been there, and in the last 20 years, we have been able to home in into them. So modern flow cytometry have been around for the last 80 years. One of the first applications was trying to quantify the amount of bacteria in milk. Back in the time, people did not want bacteria in their milk, so they developed at least a plan for a flow cytometer and counted bacteria was developed. And when I started my career, this was typically what I would see. People wanted to look at the lymphocytes, monocytes, and granulocytes, and everything else they would just call it debris. Back on those days with a 488 nanometer laser, you can see bacteria, platelets, eukaryotic cells, and perhaps what you're seeing over here are the large extracellular vesicles. Then in the last 10 years, we achieved 200 nanometer resolution with the use of violet size scatter. Then was the well, as the violet lasers became readily available, many instrumentations began to use it, including the CytoFlex LX and S from Beckman Coulter, as well as other companies. Over here, we have some just plastic beads that are 220 nanometers, 450, 600, and a bit over micron. And that allows to see small extracellular vesicles as well as everything that we're seeing before, everything in the same instrument all at once. Recently, due to technical achievements, we are now able to achieve resolution below 100 nanometers. That actually allows to see antibodies, viruses, as well as the extracellular vesicles and large vessels. I'll be talking a bit today about the Beckman Coulter CytoFlex Nano, which actually it focuses on this area between 50 nanometers and 1,000 nanometers. With such a machine, I saw, for example, a researcher being able to identify single variant cells labeled with antibody, separated from the antibody without the virus or the virus without any labeling. The CytoFlex Nano, it has a very small footprint. It's about the size of what a printer used to be not too long ago. And the two aspects that make this instrument particularly good at looking at submicron particles are the sample input, as well as the optical bench that can capture information about these particles. If you are familiar with the CytoFlex technology, you will see that the optics is held in wavelength division multiplexers. This machine has two of them that capture the size scatters as well as the fluorescence. The software is very easy to use, similar to the regular CytoFlex. You have control over the sensitivity of your detectors, as well as your threshold, and the ability to do conventional compensation. The machine comes equipped with 4 excitation lines, which are pretty generous. The 405 laser is currently the gold standard to look at these particles, submicron particles, as well as you have your blue, yellow, green laser, which they help you to look at your fluorescent detectors. You will see in the literature a lot of talk about violet size scatter as being used to identify these extracellular vesicles without fluorescence, just based on the scatter. This instrument actually has 2 detectors dedicated to the 405 laser. They call violet size scatter 1 and 2. I'm not sure what is the secret in the sauce, but one of the detectors is homing to look at the smaller particles between 40 and 150 nanometers, and the other detector is optimized to look at particles between 80 and 1,000 nanometers. You're not supposed to run particles that are over 1,000 nanometers a micron. Thank goodness nobody clogged the machine, so I don't know if people were really good about preparing their samples or if the machine actually have a way to prevent clogging. Now, something that I haven't heard people talk about this instrument is the fact that besides your violet scatters, you're also getting your blue size scatter, your yellow-green size scatter, and your red size scatter, which they are not optimal to look at submicron particles for multiple reasons. However, from the practical point of view, if you were using this instrument to look at your submicron particles, somebody who has the time and the energy and the interest, they might be able to use the different scatters to, for example, separate a plastic bead from a particle of biological origin and so on. You might be able to trigger in with a fluorescent marker. I don't know if the instrument's already been optimized for the detection of the actual exosome. The instrument runs an optimization for the detection of the signal. And we have 6 fluorescent detectors, which are pretty standard. The dyes that I have over here were some of the ones that were used, BB421, FITSI, PE, APC, RB750. I will say that some of the best results were obtained by people who label their own antibodies, and they run quality control for signal to noise ratio. And I noticed that they were using Alexa 488, Dialight 550, and Janela Fluor 646 to label those antibodies. These plot shows a peak fluorescent detection for the 6 channels that I have mentioned before. And typically what you get is for your violet fluorescence, your blue 405, and your yellow-green, you get a good definition of the 4 peaks. Then in the red fluorescence, then the smaller beads, they tend to merge together because those are low energy wavelengths. Typically, that's something that I experience with other instrumentation. So I guess that using the violet, blue, and yellow dyes, they tend to give better separation of the signal. So the optics is super important for the function of this machine. But the other part that makes this process happen is the sample loading. It's a single tube loading, which seems to be is the way that we used to do things before. However, there is a complex system to load the sample where it uses a piston pump to move the sample precisely. There is some filtering of the sheet at 5 nanometer level. There is the 405 laser for triggering and detection of the particles. And very importantly, there is an automated backflush that makes sure that none of the sample from the previous sample is in the following sample. This seems something straightforward and you can do yourself with every machine. But as we have been talking about standardization and other processes, putting it all together in one instrument ensure that your data is processed to the best possible standard. So what, for the time that this demo was performed at NEI, we have several labs walk through the core. And basically their interests were exosomes, nanolipid particles, and viruses. The software for the CytoFlex was updated and make it compatible with something called FCM pass, which is something that would be very interesting to people looking at this particle. And I learned a lot about sample preparation. One of the key points that I would like you to remember at the end of this talk is submicron particle analysis is quite different from cellular analysis, and you will have to adapt your protocols and reagents in order to get the best possible signal. So let's talk about the important things that happen in the lab. We have a demonstration. We were looking at exosomes. And one way to identify exosomes is by using annexin 5 for the identification. So, you can clearly see the noise level of the machine separated from the exosome part of the sample. Over here with annexin 5, we can actually separate the the annexin 5 positive particles from the non-exosome positive particles. Also, if you're interested in platelet analysis, you might be using CD285-8 or BLAN3 to identify those exosomes. So we have an example over here for positive expression of the marker and negative expression. One of the protocols that we ran to the machine already made it through scientific review. And while this is not my favorite image, that was good enough for the process of peer review. So, yeah. Something that people ask about, well, what should I consider when looking at bringing this instrument into the lab? It's definitely meeting minimal information standard for extracellular vesicle analysis and MyFlow. For drug developing, the instrument, according to the specifications, you have been designed to meet CFR Part 11. And it's only for research purpose. That's with respect to extracellular vesicles. Then the other part of perhaps the most interesting part of the work that was done with this machine was the analysis of lipid nanoparticles, LNPs. Just because there's such an area of interest in research today, right now they are being developed for gene editing tools. Recently in a protocol that have been able to, then is being developed for the treatment of cystic fibrosis in vitro. I believe that that work was done. And also for CAR T cell modifications in vivo, as well as the study of complex nucleic acids like circle RNA instead of your traditional RNA message. Precision oncology on, for example, how to create a vaccination for neoantigens, and in neurodegenerative diseases, as NLPs are being tested as a way to be able to cross the blood-brain barrier. So one of we have several NLPs run through the lab. This is an example of one of the experiments carried in the lab. As something that if you're not familiar with NLPs, you will be surprised to know that not all of the NLPs actually carry the load. It's actually typically a small fraction. Over here, the dye Cyte9 with Cyto9, which actually labels the nucleic acid, is used to identify the nanolipid particles that actually carry the mRNA load from the negative sample. And typically we will run that on with a titration of the dye. Something that this instrument can do is if you run calibration with beads, which it's informative and perhaps not perfect, you will be able to calibrate the scatter of your particle to the beads and make an estimate on what is the size of the particle. So in this case, you will see that one of the x-axis is robust coefficient variation for calculated sample diameter. So this gives us an estimated size of the nanolipid particles that we were looking at. However, we have been discussing a lot of technologies here today. The reality is, if you take this sample and we'll look at it with cryo-EM, for example, you will get a slightly different size. The idea could be that the cryo-EM has dehydrated the sample in some ways, but as not so obvious to everybody, if you use multiple techniques to measure the same thing, you're going to come out with different measurements. So SCM pass is not necessarily associated with the instrument. You can actually run it with data from pretty much any machine, I believe. But FCN Pass is now integrated into the Cyto Expert Nano 1.4 software. And I guess is what is being shipped with the machine. The last data that I will have the time to show you today was the identification of single Bayer particles used by a scatter alone. These can be advantageous. This is influenza A, which I guess is around 80 nanometers. Over here we have our buffer alone versus the viral particles in solution. The same sample was immunostained. So in red, what we have are the virus alone. In orange, we have the antibody that was used to label the cells. And here in this box, we have the viral particles that bound the antibody. I guess on the Now, with respect to the ability used to count particles, we have I just wanted to show a plot of the correlation between the estimated amount of beads and between the CytoFlex Nano and the CytoFlex Lex. The CytoFlex Nano is have a R-square of 0.9992, which is a little bit higher than the regular CytoFlex. These wanted to mention the point that, well, having an instrument dedicated particular for nanoparticle analysis is an investment, but at the same time, in order to get a bit more extra ability to count the particles and determine the size is part of the cost of, working in such a small part of the space distance. So what I think that we should keep in mind about nanoflow today, extracellular vesicles, nanoliquid particles, and viral vectors are no longer the future of medical care today. It is actually being developed into the precision medicine of the near future. Some of the techniques to prepare the samples for the machine, besides involving ultracentrifugation. Some of the best data actually involve cesium chloride centrifugation, which are techniques that are common for some work, but to me, they were new to me as a flow cytometry professional. But they can actually were necessary to produce a better readout. One of the greatest things about submicron sizing is that FCM technology, which is based on my theory, might provide one of the first examples where we can actually standardize measurements from one machine to the other and being able to get measurements that are the same across different instrument platforms. Thank you very much for your time, and I look forward to answering any questions now or the later event. Thank you. Thank you very much.
Okay, well, that wraps up our program. Just some last little closing words here. You know, these things. Our next location is Pub 70, which is the happy hour sponsor, Beckman Coulter. Thank you. And I hope everyone is able to come and enjoy a beer or wine or whatever. There's going to be tickets available for all of you when you get to the door for drinks. And then there'll be food available starting at 5:30. I just want to say some last closing words. First of all, I appreciate and thank everyone for coming here today. We would not have this meeting if it wasn't for your participation. I want to give a heartfelt thank you to all of the speakers, the panelists, the poster presenters, and for sharing their work and their experience and their time. Their contribution is what turns this into an event, from a schedule into an event. I'm also deeply grateful for all of our sponsors and industry partners and their generous support throughout the day. Without them, we would not have this meeting. And finally, thank you to all the attendees for your questions, conversations, and enthusiasm. The Northwest Flow Cytometry Northwest Flow Cytometry Society exists to serve this community. And your participation is what drives us and keeps us building this annual event into a yearly thing that I hope that all of you will continue to attend and participate in. With that, we're going to officially close out the program, and we will hopefully see you soon at Pub 70. Thank you, everyone.